சார் பேச கேக்குதுங்களா? ஹலோ. வெயிட்டிங் ரூம் இருக்கட்டும், பிரச்சனை இல்லை. ஸ்டுடியோ மாதிரி பிரச்சனை இப்போ பார்த்துக்கிறேன். உங்கள் பேரில் வந்து மட்டும் நீ ஒன்றும் அட்மிட் பண்ண வேண்டாம். நான் அப்புறம் ப்ரோக்ராம் ஆரம்பிச்சு கூட அட்மிட் பண்ணிக்கலாம். வெயிட்டிங்கே இருக்கும் என்ன.
Hello, hello. I'm checking. Sir, கேக்குதா? Please, sir. Sir, system. Hello. என்ன இந்த பின்னு நம்ம கேட்கலாம் சரி பண்ணு Mic check. Hello. 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 Mic check. Hello. Hello. அந்த மிக் கழிச்சல ஆடினா ஆடியோ வீடியோ வீடியோ நீங்க அங்க பண்ணி
informative, and entertaining. We don't dream, we drive ideas for life. Good things happen to those who write with a sense of direction and stubborn determination. And here comes the knowledge of emotional molecules that would melt into your brain, into your dreams and empathy. As we celebrate our success and as we move to take the first wheel of life, let us pause for a while and put ourselves in most good persons of knowledge Almighty. I request our students to be them in prayer.
இதயம் மருகில் அமர்வாயின்றே இறைவா இறைவா வருவாயின்றே இதயம் மருகில் அமர்வாயின்றேன். உன் தேடல் நீ, என் தெய்வமே. நீ இன்றி என் வாழ்வு நிறமாறுதே. உன்னை மனம் தேடுதே. நீ வழிகாட்டுமே. உன் தேடல் நீ, என் தெய்வமே. நீ இன்றி என் வாழ்வு நிறமாறுதே. உன்னை மனம் தேடுதே. நீ வழிகாட்டுமே.
Students while watching the pursuit of Lord Almighty. In the beginning, there was nothing. God said, "Let there be light." Over the next 6 days, He created the universe. Let us start with light, and over this day, we will see this universe. It is the dictionary that is to light the light. I request respected Reverend Father Lecter, respected Reverend Father Principal, respected Dr. Varadeshan, Emeritus Professor MS University, respected Dr. Raja, Department of Special Department, and a student volunteer to Father. We welcome you now and always with sincerity because we will be with you right through. We welcome you now and always with sincerity because we will be with you right through. Even though you may be miles apart, your memories will be made close with our heart. In an occasion like this, you deserve a special person to welcome the special of you. Dr. Raja, the convener of this international conference, is an ardent researcher and able teacher. His illustrious research career spans for over 2 decades with many publications and projects. He has started contributing to the universal entropy since 1981 and the empathy to our department since 2019. We are really proud of you, sir. Let us, with loud and of applause, welcome our
the beginning of the before I also feel it is my problem to
It's my pleasure to welcome all of you to this international conference organized by the Department of Zoology, St. Xavier's College, entitled Recent Trends in Aquaculture. It is an opportune time to welcome Reverend Dr. Benji Garo, the Director, who is the head of the several institutions, the heart of the community and eminent educationists, and a renowned social reformer. Welcome you, Father. We extend a warm welcome to our beloved principal, our Father Mariadas, yesterday. Father has been constantly and consistently encouraging and enabling us to organize such academic programs from the very inception of hosting the conference. Father has guided and supported us to take this initiative. Once again, thank you, Father. We have to welcome Dr. Salami from DCCSR, Department of Animal Science, and the illustrious alumnus of our Department of Zoology. Sir, thank you for accepting our invitation and we are unanimously coming up with the decision that we must invite you to the keynote address because of the commitment to have been doing in the field of aquaculture. Welcome, you, sir. Yes, Raja, Assistant Professor, Department of Zoology, Bangala Arts and Science College, Coimbatore. Dr. Ram for Assistant Professor, Department of Zoology, Sciences, Joseph College, Autonomous Bangalore. Dr. K. Marimuthi, Professor, Anis University, Malaysia. Dr. Gerald Andoni, Aquaculture Expert, Sudan. Dr. C. Stalin, Assistant Professor, Department of Zoology, Euro Arts and Science College, and Dr. T. Bharati, Assistant Professor, Department of Zoology, Sir. Jagaraja College, Chennai. All of them have already accepted our invitation and being with us here. Welcome you all. And then to welcome Dr. Bhuti Sam, Education Coordinator, St. Xavier's College. He has been continuously encouraging us to do such a program. Welcome you, sir. I fondly remember and welcome Dr. M. Hanifa, founder of Center for Aquaculture Research and Extension, Department of Zoology, for his tremendous contribution to this aquaculture field, especially marine culture. And further welcome Dr. Sivaram Lingam, Dr. Narayanan, and Dr. Victor, Professor of Department of Zoology, to also in the field of aquaculture. I remember everyone with a heartfelt thanks. I extend a heartfelt welcome to faculty members of the Department of
ഞാൻ
Zoology.
എന്റെ PPT നിനക്ക് ലാസ്റ്റ് കോപ്പി ആയിട്ട് അയച്ചു തരാം. ഞാൻ ഓൾറെഡി അയച്ചിട്ടുണ്ട്, ഒന്നുകൂടി അയച്ചു തരാം കേട്ടാ. ഡൗൺലോഡ് ചെയ്ത് വെച്ചോ.
Welcome Dr. Raman. Always showcasing the passion to pursue excellence. Welcome, sir. I also welcome Dr. Noble Kalimula and extend a heartfelt welcome to the participants from both online and offline for your overwhelming response and the response to support the online conference. Faculties, research scholars, students of different institutions, I welcome and wish you all enriching experience. I welcome all the teaching students of students and research scholars of our department. Welcome you all, my dear students. The objective of this conference is to enkindle research interest among the budding research scholars and acts as a platform to showcase the contemporary research. It makes an important contribution by highlighting the significance, value, and credibility of aquaculture research, with special emphasis to regional studies. It is also an important attempt to create a platform to discuss the issues in aquaculture development, especially challenges into the global warming and water pollution. I welcome and thank our college faculty and various friends for your support. I wish you all an enriching learning experience. Thank you, one and all. Thank you.
Thank you, sir, for such a warm welcome.
സൗണ്ട് കേൾക്കണ്ട, ലോ വോയിസ് ഉണ്ട്.
Stable, capable, all together with humble. Defines our reverend father principal, the prime mover of Xaverian institutions. A reverend father is an epitome of duty, honor, courage, commitment, vision, tenacity, and an indomitable spirit that inspires everyone around him. His eyes have a touch of an ice and make us feel brand new. He is very good orator and makes ours melt away. Now, hand of applause, let us all welcome our reverend principal to deliver the presentation. Respected moderator, Xavier institutions, respected Dr. A. Paralekhan, our illustrious alumnus and UPCPSR Fellow, Department of Animal Science, MS University, respected Dr. Raja, HOD of Zoology, all the professors, good morning to you all. As I have very important meeting, I have to leave immediately because around 10:30. I'd like to wish you all the best at the outset for organizing this wonderful international conference. You have a gallant and eminent speakers, because Dr. Palavesan, a wonderful person, a person who shows lots of interest in our institution. You know, he is our alumnus. I have been interacting with him for the past 2 years, more than 2 years. Whenever I interact with him, I really get inspired the way he has attached himself to the institution. His attachment, sense of belongingness to the institution is very amazing. You will definitely listen to him. And I also know that Dr. Harold Anthony, Aquaculture from Sudan, he was also our alumnus in the year 1992 to 94. He did his MSc zoology here when I was there in the hostel. He was a student. Wonderful person. I think his sister joined and he left. And of course, there are other eminent speakers. I wish all of them very well to interact our students with our students and other students from other institutions. Therefore, I wish you all the best. Have a wonderful day. Have an interactive session. Have a productive session, so that we equip ourselves with more skills and knowledge, so that we will be able to establish our survival and stability in the society. I wish you all the best. May God bless you all. Thank you. Thank you, Father. We are really grateful to you, Father, for sparing your very valuable schedule to be with us in this occasion. Your views are critical to us. Now it's time to release the abstract. I request our Father Rector to release the abstract, and request our Father Principal to receive the first copy of the abstract.
So congratulate all the organizers. And the questions to take you on. Productions. It's not as much as as organic productions, like in plants Second one is to make this is making as a culture of the people. Any change would happen and the brilliant one only when it joins the culture of the people. I feel that organic interest in organic things now has become a culture of people. You have to observe Italian conference, quite many people presenting at the Sitara conference, many people came. We have, for example, when there is a town we had people visiting things, and then many people went. Find that now that is becoming a culture of our faith. Not always we relying on the allopathic medicine, now we need to go for alternatives. And alternatives are found also in allopathic productions. One thing is that people have to be given that culture of making it as part of us. I'm sure that such research, such conferences will give us an orientation, a direction for people to make it as part of our culture. And very relevantly, you have taken this topic for the international conference. I appreciate all of you, dear professors and students of the Department of Zoology of our college. God bless you all. Thank you.
Thank you, Father, for gracing this function and sharing your views with really engaging delivery. Thank you once again, Father. Now I request our Reverend Father Rector to honor Dr. Palavesam, the chief guest of this international conference. Life is busy. Serious makes it easy. We switch on the best in you to face the world outside. Seeing this one thing changes everything. At St. Saviour's, we are many, but we are one. We do what we love and love what we do. We look alone, but never feel lonely. We want to please, it is our time. We did it together. This is the way we move at St. Saviour's. Let us all welcome Dr. Sandra Kumari, the co-organizing secretary of this international seminar, to propose the vote of thanks.
On the dais and off the dais, respected professors, scholars, students, guests, members of the media, ladies and gentlemen, and above all, Dr. Palavesam, a very pleasant morning to meet to one and all. I deem it as a great honor and privilege to propose vote of thanks on behalf of Department of Zoology of St. Saviour's College on this occasion. First and foremost, I profusely thank Reverend Dr. Vendi Jerome S.J., Director of this great temple of learning, for having graced this occasion by sharing his precious time with us today. Reverend Father Rector, he is being always extends his moral support and guidances in all the academic endeavors of us. His unstinted devotion helps the department to uphold its vision and vision. I wholeheartedly thank you, Father, for presiding over the inaugural function of a historically important academic event organized by our department. A big thanks to you, Father, for your noteworthy words of blessings and for your kind words. Next, we are blessed to have Reverend Father Maridas SJ, the principal of this heritage institution, amidst us today on this occasion. I wish to express my gratitude to Reverend Father Principal, whose presence has filled with us with great pride. As the academic head of this esteemed institution, he is a motivating force and a source of encouragement to all of us. The solid support blended with the encouragement and motivation always have a positive impact in our every academic effort of the department. It has resulted in achieving remarkable milestones. I sincerely thank you, Father, for your felicitations. I wish to express my gratitude to Dr. Professor Palavesam, UGC DSR Fellow, MS University, for spending this valuable time with us. I thank Father Rector for being kind enough to release the abstract, and I thank Dr. Palavesam for receiving it. And I thank Dr. Raja, Assistant Professor from the Arts College, and Dr. John Paul from St. John's College, Bangalore, and Dr. Shanmugude, Assistant Professor from the Arts College. I thank every one of you to be present here. Thank you.
Displayed, we will do it later. Let us honor other dignitaries. I request Dr. Palavesam, Emeritus Professor, MS University, to honor Dr. Raja, Assistant Professor, College, with a small I also request Dr. Palavesam to Dr. Sanna, Assistant Professor, Kudunar Arts and Science College, Coimbatore. Request Dr. Palavesam to honor John Paul, Assistant Professor, St. Joseph's College, Bangalore, SR and Toronto. Thank you, sir. Thank you very much. Thank you, everyone. Thank you, everyone, for your patience and attention. And now I request Dr. Mabel Parimula, Assistant Professor of our department, to introduce the keynote speaker, Dr. Palavesam.
Dr. Palavesam. Dr. Palavesam is a UG DSR faculty fellow in the Department of Animal Science, Manoormani Sundarkar University, Tirunelveli. He is blessed with 25 years of teaching and 28 years of research experience. His area of specialization includes fish nutritional biochemistry and physiology, immunology, probiotics, and microbial ecology. He has guided around 34 PhD candidates and 3 postdoctoral fellows with 8 more ongoing PhDs, and has also the credit of guiding 100 MPhil projects. He has to his credits 180 research publications in the national and international journals and proceedings, and 3 book chapters with more than 1,600 citations, 21 H index, and a cumulative impact factor of 146.3. He has served as a convener of PhD Viva Voce examination for many universities in India, including Kerala University, Bharatidasan University, Alagappa University, Pondicherry University, Madurai Kamaraj University, Annamalai University, VIT University, and Periyar University. He is the recipient of many prestigious awards. Tamil Nadu Senior Scientist Award in 2016, Recognition and Citation Award from the Society for Advancement of Biological Sciences, Excellent Paper Award, Best Faculty Award, MDT Investigator Award, Academic Excellence Award, UNESCO Biotechnology Fellowship Award, UGC BSR One-Time Grant Award, and at present he is a UGC BSR Faculty Fellow for the years 2021 to 24. His significant contributions to the field of aquaculture includes formulating nutritionally balanced, cost-effective, and stable artificial feed for fattening of baby rock lobsters, screening, characterizing intestinal probiotic microbes, and developing cost-effective feed probiotic for enhancing growth and immune resistance in shrimp, identifying more than 60 probiotic microbes from marine resources through 16S RNA sequencing and submitting in GenBank, screening, characterizing, and identifying antifouling compounds from potent symbiotic microbes isolated from the marine environment, identifying eco-friendly antifouling compounds from fractions of seagrass, and identifying 5 marine keratinogenous yeast species for their carotenoid production. He also has rich administrative experience, beginning as a coordinator of UGC staff program, principal in charge of MSU Constituent College, Kanyakumari, professor and head, Center for Marine Science and Technology, special officer, Directorate of Vocational Education, MS University, professor and head, Department of Animal Science, MS University, Director, Center for Internal Affairs, MS University, Dean, College Development Council, MS University, to the very recent registrar in charge of Manonmanim Sundarnar University. He has undergone special training programs in US, Greece, and Dubai, and has had the privilege of visiting countries like Singapore, Malaysia, UK, Thailand, China, Abu Dhabi, Oman, Doha, Sri Lanka, and Ethiopia. He had the honor of completing 12 UGC major research projects worth rupees 238 lakhs, 12 consultancy projects worth rupees 19 lakhs, and other special grants worth rupees 167 lakhs. He has been invited for 91 lectures, 1 radio talk, organized many conferences, participated in around 50 conferences, seminars, workshops, and has been an active member of the Senate, CH Doctoral Committee, RUSA Committee, Flying Squad and MSU exams, Board of Studies Chairman, IQAC, and UGC Plan 11 and 12 Committee member. We are honored to have such an eminent personality among us, and may I now request Dr. Palavesam to deliver the keynote address. Thank you.
Project
Morning to one and all present here. At the outset, I would like to thank the Reverend Father, Rector, Principal, Vice Principal, and also the faculty members of the Department of Zoology of St. Xavier's College, the convener of this conference and organization secretaries and other members of this conference for giving an opportunity to come over here and share my experience, what I have gained during my research in the area of aquaculture. As far as the theme of this conference is concerned, it is the need of the hour. And we are doing wonderful research in aquaculture. And our research should not be confined to a particular area. As and when we have to refresh or we have to strengthen our idea, strengthen our knowledge in that particular area. Within this context, organizing such conference is inevitable one, and it is also a welcome approach. And the title of the conference is Recent Trends in Aquaculture. My talk will be on, first I will give you a preliminary idea about what is aquaculture, what is the significance of aquaculture, and what are the various components which is involving in the aquaculture practice, and also the hurdles in each and every aquaculture components, what are the hurdles are there, and how the hurdles are handled. Based upon the availability of technology from the olden days up to the recent past, new technologies are being innovated, and those technologies are implemented in the aquaculture operation so as to get the sustainability and maximum production. And in this brief introduction, and what you are seeing is just to refresh yourself that aquaculture is culture of aquatic organisms of desirable character, that you have to add that in majority of the books that culture of aquatic organism in a confined environment from dry or pickling stage up to a marketable size is called aquaculture. Now, I request this audience, you have to add one more thing, culture of aquatic organism of desired character. That is very, very important. Based upon the character only, and we are selecting the species and we are culturing it. Desired character in a confined environment from dry or pickling stage up to a marketable stage is called aquaculture. And as far as aquaculture is concerned, there are various components, and you are all aware that the hatchery and then feed production and then grower system, harvesting and marketing. These are the various components as far as aquaculture is concerned. Next slide. Now, and as far as fishery production is concerned, as a fishery, there are two major sectors. One is from the natural fishery resources, and another one is from aquaculture. And if you see the contribution of fishery production of natural resources, in the earlier days, the contribution of fishery production from the natural environment, that's the natural fishery resources is very high. And after certain duration, and if you see that there is stagnation in the natural fishery production, what you are seeing in the below border is the fish production from the natural environment, that is the fishery production. And if you see the growth of the aquaculture in the upper panel, just is a target, this shadow that indicated the contribution of aquaculture production. And if you critically view this slide, you can able to understand what is the significant contribution of aquaculture in fish production and what is the need. Why this we are all doing the aquaculture research and we want to enhance aquaculture production and so on and so forth. And as far as fishery production, there is a stagnation. At the same time, if you view the population of our country, before 20 years back, our population is 1 billion. Now, what is 7.9 billion, our world population? Just imagine that there is an 8-fold increase in the population, but if you see the fishery production, there is a stagnation in the fishery production. And there is a vast gap between the what actually we are producing, what's the demand. And the demand is very high, the production is stagnated. It is in this context, aquaculture is emerged as an alternative sector to fishery production and also the agricultural production to meet the protein-rich food material. Therefore, aquaculture industries is emerged as the multi-billion-dollar industries producing food materials, protein-rich food material at a reasonable cost, and also it contributes to the national economy. Once the majority of the countries, the economy of the particular countries is being determined by the aquaculture production. It is in this context, the aquaculture is considered as the most significant one. Next slide. Next slide. Okay. As far as significance of aquaculture, here, so protein food materials to the undernourished population, and apart from that, this aquaculture industry is producing employment opportunities to the skilled and unskilled laborer, and it is also earning foreign exchange, and it is also rehabilitating the fishermen community and those who are deprived of natural fishery product. And it is in several sense, this aquaculture is considered as the most significant one. Next slide. If you see the scenario of aquaculture, as far as FAO statistics is concerned, nearly about in terms of resources in our country, we are having 29,000 kilometers of reservoir, and then 3.1 million hectares of reservoir, rivers and reservoirs, and 2.35 million hectares of ponds and tanks. In the simple sense, we are having enough resources in terms of rivers, reservoirs, tanks, and ponds and food. And if you really take the statistics and how much area we are actually using for aquaculture operation is very less. Yet, majority of this water bodies has to be explored for the development of aquaculture system. And next slide. If you see the aquaculture production, worldwide aquaculture production, and this China is contributing topmost. China is the topmost country which is producing the maximum aquaculture product, followed by India. We are the second, and if you see this, if we are standing second, if you see the production, there is a vast difference between the production of China and our country, and yet we have to take a lot of efforts to enhance our aquaculture production. And yet, as far as available statistics is concerned, and we are the second largest producers of aquaculture. Next slide. As far as global scenario of aquaculture is concerned, and the FAO statistics indicated that 567 species are characterized and registered for the aquaculture operations. And out of these 567, and both fishes, crustaceans, amphibians, and aquaculture invertebrate and marine and freshwater algae, and various species, they are contributed in various percentages. And but in recent estimate indicated that more than 600 cultivar species are registered in FAO aquaculture. And those species are cultured from the available freshwater, brackish water, and marine environment with the input of sophisticated technologies. And so far, what I have given you is a nutshell that what is aquaculture, what is the significance of aquaculture, and what is the scenario of aquaculture as far as the Indian scenario as well as the international scenario, and how much aquaculture species are available and registered so far, and which are highly suitable for culture in the aquatic environment. Now, next slide. This slide shows that in various phases, what are the problems we are facing as far as aquaculture is concerned? What are the problems we are facing is the sense that water quality, the immediate environment in aquaculture is the water quality. And when the water quality is deteriorated or it is deviated from that of the optimal level, and there poses some problems, and that will ultimately affect the food intake, growth, and as a result, what will happen? Disease outbreak will occur. And when the disease outbreak occurs, it is expected not only to destroy the water quality, but also to the soil quality and other parameters. Therefore, these are all the problems. The problems in the sense water quality, soil quality, food intake, disease outbreak. Okay, and these are all the various problems which are encountered in the aquaculture operations. And how we are approaching this problem, whatever the problems we are facing, how we are approaching this. In the earlier days, various types of chemicals have been used. You are all aware, you might have studied from your literature that various types of chemicals and also the various types of antibiotics and other materials are used to combat the disease outbreak and also the water quality management and soil quality management. And those chemicals and antibiotics, when we are using at more than its threshold level, and it is also again posing various problems. That is why, and aquaculturists all over the world focus their attention to replace these hazardous chemicals and also the burnt antibiotics uses in the aquaculture operations. And there from that point onwards, there's a high-end aquaculture research starts. As far as high-end aquaculture research is concerned, the research should focus on the particular issues and it should yield some solution to solve the issues. And that is the thing for the advanced research as far as aquaculture is concerned. Next slide. And in my talk, next slide. Yeah. In my talk, I am going to tell something about what are the various more recent techniques which are being employed to address the issues which I have just narrated earlier. Okay. Now, as far as probiotics, you might have heard this is one of the hottest area of research as far as aquaculture is concerned. Now, when we are talking about the probiotics are nothing but the live microbial cells. When we administrated in an adequate amount, and when it is entering into the host system, it will positively affect the host system, and it will favor the host system for the better performance and to display the better growth. And ultimately, what we are getting is the maximum production with the sustainability. And therefore, with live microorganisms, and we are enhancing the production. And you may ask the question whether these probiotics are used only for enhancing the production. If various probiotics are there, water probiotics, feed probiotics, and soil probiotics. And water probiotics will take care of the water quality parameters. If there are any deviation in the water quality parameters, that's water probiotics will take care of this. And then feed probiotics, the feed probiotics is the growth promoters. And when we are administered along with the food materials or feed as additives, when it is entering into the food system or gastrointestinal environment, there it performs the better functions. It will accelerate the digestion process and it will synthesize the various amino acids and fatty acids, and it will also produce some antimicrobial peptides. By doing innumerable functions that will favor or that will help the whole system to lead a better life. And as far as soil probiotics is concerned, and you know what is the soil? If the soil is contaminated or enriched with particular nutrients, for instance, in aquaculture operation, we are using the feed of high nitrogen content. So the protein. The protein is nothing but the nitrogen. When a large amount of protein is accumulated in the form of nitrogen and in the pond bottom, that will deteriorate the pond bottom and it will lead to the various hazardous process such as the algal bloom, and it will indirectly affect the various water quality parameters. Therefore, it has to be taken care of. The soil quality parameter has to be taken care of. For that also, this probiotic techniques are being employed for there is a nullifying the hazardous effect due to the soil quality, soil parameters. And it is in that sense, various probiotics microbes are used, to solve the issues and to enhance the protection. Whatever may be the issues we are solving, it should ultimately it has to be directed towards sustainability. Sustainability is the sense. Therefore, the more recent techniques which is widely used in the aquaculture system is the probiotics. Next slide. This is the overall views. As a researcher, we should know what are the microbes we are using as the probiotics. When it is entering into the gastrointestinal environment, what are the functions these probiotics are performing? And we should have some knowledge. Then only we can take the appropriate microorganism to solve the particular issues. Therefore, here, when whatever may be the probiotics, when we are using in this aquaculture system, and when it's entering into that system, it performs nearly about 6 different unique functions. The first and foremost function is the adhesion, enhancing the adhesion and the colonization of beneficial microbiota in the gastrointestinal environment and also in the aquaculture system. That is one of the first and foremost functions being performed by this probiotic microbe. This is the first one. And second one is this epithelial variability. And you are all aware that in the living system, each cell has its own permeability function. Am I correct? And it will allow certain macromolecules to entering into that cells, and that will be more helpful for the cell function. And that is being regulated by the permeability of the cell membrane. When the permeability of the cell membrane is affected, and this abnormality may occur, and the metabolic disorders also may occur. And how these probiotic microbes, if you see that these microbes in the sense, and we are talking about microbes, but we are forgot the host system. And another one, these probiotic molecules are also associated with the phagocytic activities. And I will show you some slides there how these probiotic molecules is activating the phagocytic activity. And therefore, by doing 6 different significant functions, and these probiotic microbes are performing wonderful functions inside the host system and also inside in the aquatic environment. And therefore, it is giving this benefit to the host system, not only benefit to the host system, and it is maintaining the sustainable environment for the simultaneous for our concurrent production into the system. And this is an important area of research as far as aquaculture is concerned. Now, each and every one, if you see if you visit any research institution, you can get that one or two students and they are involving in the probiotic research. And this is a more recent research as far as aquaculture is concerned. I can cite one or two examples and how these probiotic products are acting on that. This is a EV, this is a hatchery probiotic. Hatchery probiotics in the sense that there are two different water probiotics and feed probiotics. If you want to make the water quality, and you can use the water probiotics. If you want to enhance the feed efficiency or feed consumption ratio, and you can use the feed probiotics. And therefore, either it is a water probiotics or feed probiotics, and you can use in the hatchery system as per your requirement. Okay, now you may ask the question whether we are using the single microbes or more than one or two microbes. It is the single microbes. If the single microbes, some of the single microbes can perform just various functions. And we characterize one microbe is called Bacillus cereus from the gastrointestinal environment of Pseudomonas monoda. And you will monitor that, and those single microbes, Bacillus cereus, are capable of producing protease, amylase, lipase, and chitinase, 4 enzymes. And the 4 enzymes, except chitinase, and all the 3 enzymes, they are synthesizing at the maximum level. And thus, if the single microbes is enough to meet out your requirement, you can use the single microbes, or it is a polymixture of microbes, and a polymixture of microbes based upon your requirement. In these specific cases, Bacillus acidophilus, Bacillus subtilis, Bacillus riciniformis, and Nitrobacter, Aerobacter, and Saccharomyces cerevisiae. This is a polymixture of microbes, and this polymixture of microbes was used as it's a water probiotics to enhance the water quality parameters. In the same way, this is a hatchery feed probiotics, Lactobacillus, Bacillus, and also the Saccharomyces cerevisiae, and they are used. They are using as the feed probiotics as far as hatchery is concerned. Next slide. And another one important this is ABL, ammonia binding inoculum liquid. And this is also a probiotic mixture culture. You are all aware that ammonia is the major nitrogenous excretion as far as aquaculture is concerned. And if it is accumulated in the aquatic environment more than the threshold level, and you know that what is the effect of the accumulation of ammonia, and it has to be taken care of. And therefore, based upon this concept, a product is being developed that is the ABL, ammonia binding inoculum liquid, and this is contain nothing but the nitrosomonas and nitrobacter, and those bacteria will take care of the conversion of ammonia into nitrate and then nitrate, which in turn ultimately it will be used for the fertilizer in that one. This is the second commercial probiotics, and we have also characterized various probiotics. You can skip off. You can skip. Yeah. Okay. Now this is a hatchery. I have cited 2 examples for hatchery, and also this is a crowd system. In the crowd system, and here wherever the stage you want to use this probiotics or whatever may be the more recent techniques, you should aware that and what is our need and what's the problem actually going on. And as a zoologist and as a biologist, you should know, and the growth is being governed by both the abiotic and the biotic factors. And this, what are the abiotic factors and biotic factors that is governing the growth of these organisms? And based upon that, and you can use the probiotic microbes. Here also again we are comparing water quality and nutrition is the major factors that determine the growth as the growth system, similar to that of the hatchery system. Here we are giving in growth system, we are giving much concentration on growth. And feed probiotics, as far as growth system is concerned, feed probiotics playing an important role when compared to that of the water probiotics. This is the growth system. What are the hurdles we are facing is the larval mortality, feed related waste accumulation, disease outbreak, slow growth factors and water and sediment quality, deterioration of water and sediment quality. After ascertaining and what you are experiencing in your own pond, based upon your hindrance, you are basic, and you can use the appropriate probiotic to solve the issues. Okay. Here the lactose act is a commercially available probiotics. This is a water as well as a feed probiotics. The lactose act can be used both water as well as a feed probiotics. From the word itself, and you might have understand that the lactose act is the lactic acid bacteria. That's a major component in this probiotics is a lactic acid bacteria. Okay. Okay. Yeah. Just now I told you that in our laboratory, and we characterize the Bacillus cereus, one of the important and significant microbes. These microbes hold this majority of its activities, protease, amylase, lipase, and kinase activity. We characterize these microbes, and those microbes we have given in the two different forms in the sense lyophilized powder as well as the low-cost probiotic powder. And if you refer any literature, and you can get how we can prepare a lyophilized powder or low-cost probiotic powder. And we prepared in the two different forms, and we have tested this particular probiotics is showed better efficiency in enhancing the immune parameter and also the growth parameters. Okay. This growth parameters in the SGR and FCR is the specific growth rate, and FCR is the feed conversion ratio. And these are the two key indicators as far as growth system is concerned. And once you are enhancing the SCR and the FCR, on hand you are getting the more production, and the FCR value is also high. And indirectly, and you are enhancing the feed efficiency and the efficiency of the organism for performing better growth. And alternatively, what you are also doing is you are reducing the feed-related waste accumulation in that particular environment. Whatever the feed you are offering to the system is 80%. The feed efficiency is more than 80% or 90%. Obviously, less than 10% only will be accumulated in the pond bottom. Instead, if the feed efficiency is only 50 to 60%, and what is the ultimate accumulation is 40 to 60%. And therefore, if you want to accelerate the growth as well as the feed efficiency, you can use appropriate probiotic microbes, and you can solve the issues of poor growth performance and also the poor feed efficiency ratio. And we have preferred that there is an enhancement in nearly about 34% of FCR and 20% of SCR. And this is a single microbes. If you admixture the polymicrobial culture, still you can get better result in this. Okay. It's a low-cost probiotic powder. Okay. Yes. Now, when we are doing some research, always we are aiming at some better one. Okay. Now, probiotics is performing its own function, and the researcher is to find out that how we can enhance the efficiency of the probiotics. And the probiotics is entering into the whole system, whether it is inside the whole system or inside the environment, aquatic environment, it is performing its own function. And if you want to enhance the performance of this probiotic microbe, what is the alternative? That is called the prebiotics. Prebiotics are nothing but the inert material. When it is administered into the whole system, it will not undergo any digestion process. Okay, it will not undergo any digestion process. It will available as a substrate for the probiotic microbes to perform its own function or to enhance its efficiency in production. Such materials are called prebiotics. Now, prebiotic research is also getting momentous interest in the recent days. So, as we enhance the efficiency of probiotics, these are some prebiotic materials. It's an inert material, indigestible inert material, which will not undergo any digestion process. Such materials are widely used as the prebiotics. Okay. Now, our day-to-day activities, we are using some prebiotics. Can you able to tell something? In day-to-day activities, routinely, almost in every day, we are taking prebiotic without our knowledge.
Ginger, garlic.
Ginger and garlic is the immune modulator. Okay. So, third is the probiotic, only probiotic. I am talking about probiotic chapter is over. Now we are in the prebiotic. Banana. Banana is the fibrous materials which are present in the banana. It is acting as the prebiotics to enhance our own gut microbial diversity, gut microbiota. our own microbiota. Banana is a merely ripened banana. Merely ripened banana, not fully ripened banana. Fully ripened banana, and then you know the composition and everything will be varied. It's a nearly ripened banana. It is having majority of the fibrous materials, and those materials are very much essential for enhancing the efficiency of our own gut microbiota. Okay, next slide. The prebiotics. And then another one step, how we can enhance another one step in the sense, functional food formulations. Functional food formulation in the sense, as a biologist, you should know and what are the various components we are using for the preparation of aqua feed. And each component or each feed ingredient has its own inherent capacity so digestibility, in the sense of digestibility, own digestibility. If you want to enhance the digestibility, again you can go for you can take the bioconversion process. That is nothing but the bioconversion. As a bioconversion in the sense, if you take the protein material has its own digestibility of 60 to 70%. Here again, 40 to 30% is available as the waste material, undigested protein materials. If you want to enhance the digestion process, you take the proteolytic microbes, characterize the proteolytic microbes, and you do the fermentation process. It's an open fermentation or closed fermentation process. And during that process, what is happening in the sense that whatever may be the proteins that is converted into amino acids. And previously, before bioprocessing, if you see this 60% is 60 to 70% is protein digestion, and after this fermentation process, and you definitely you will get nearly about more than 80 to 90% of digestibility. Therefore, this technique is called bioconversion of feed ingredients. Here, again in bioconversion of feed ingredients, this is also a recent research as far as aquaculture is concerned to enhance the feed efficiency and also to formulate the functional food formulations. And you might have heard that the functional food formulation. Functional food in the sense, functional food materials contains, there is a easily digestible food material along with the cyst and also the microbial biomorphs, along the cyst as well as the microbial biomorphs. That is called the functional food. When we are preparing the feed along with the functional bioconverted functional food materials, and obviously you will get the maximum production. And what is FCR? How we are calculating the FCR? The amount of food we are given to the system or given to the organism divided by the growth displayed by the organisms. That is by relating the amount of food given and also the growth displayed, and we are calculating the FCR. And here amount of food given in the sense this food should be the food materials whatever we are offering to the species, it should be digested in a proper manner, and the digestibility coefficient must be more than 90. Then only you will get the FCR. Okay, and the FCR in the sense whatever the feed we are preparing under the laboratory, and majority of the study, majority of the thesis, and I have witnessed that FCR is 3.2 and 4.5. 2.2. 2.2 in the sense 2.2 gram of feed is required to produce 1 gram of fish. 4.2 in the sense 4.2 gram of feed is required to produce 1 gram of fish. Okay, this is the concept of the FCR. And when we are taking that is a functional food, the FCR will be reduced to 1.2 or 1.5, 1:1 FCR ratio. Whatever the feed we are importing from the international countries, and they are displaying 1:1 FCR. What is 1:1 FCR? 1 gram of feed is required to produce 1 gram of fish. Here, what is the waste? Waste is zero waste. Okay, that is why all the aquaculturists are only concentrating on the international imported food material. Still, other country we have to explore lot to reduce the FCR ratio of the feed. And for reducing the FCR ratio, there's a more recent techniques which we are adapting is the probiotic techniques as well as the functional feed formulations. Next slide. So bioconversion, this is a specific examples and how whatever the feed materials and you are using. So prawn-led base. Prawn-led base is also one of the major ingredient as far as shrimp feed is concerned. And if you convert the prawn-led feed, prawn-led the materials இருக்குது chitin. And chitin, if you want to digest the chitin, what is the microbes we have to use? Chitinolytic microbes. The microbes capable of synthesizing chitinolytic microbes, if you use along with the chitin, and you add some amount of chitin microorganisms, that will take care of digestion of the chitin. Okay, and what are the end product is the N-acetylglucosamine that is readily available. This is the form of glucose. What we are taking is the glucose is like that. N-acetylglucosamine is readily and easily assimilated with the aquatic organisms, in particular shrimp for the synthesis of its own exoskeleton. Okay, therefore, I have cited one or two specific examples and how we can use this probiotic microbes for the conversion of this bioconversion product. Next slide. The bioconversion of sugarcane bagasse and then bioconversion of the prawn meat waste. Next slide. As a chicken intestine waste. Chicken intestine waste is also widely used now. A proven technology has been emerged by utilizing 100% chicken intestine waste, and they have succeeded in bringing nearly about FCR of 1.4. This is one commercial company. And chicken intestine waste. This technique is only possible because of this bioconversion process. Next slide. The sugarcane bagasse and this is a sugarcane bagasse is cellulolytic microbes. This is exocellular. This cellulolytic microbes can capable of synthesizing the enzyme that is exocellular hydrolase and beta-glucanase and cellulobase. And three different enzyme systems are responsible for the digestion of cellulose materials and when we are incorporated in the feed. Okay. Also enzymes alone we can use. This is a microbial enzyme. This is a exoenzyme. Addition of exoenzymes in the feed is also gaining momentous importance so as to enhance the feed efficiency. This is a exoenzyme. And you can take the microbes, produce the enzymes, and in the cheaper rate by using the raw material, cheaper raw materials. And if the production cost is very less, and use this enzyme as a exoenzyme along with the feed as a feed additive to enhance the feed efficiency. Okay, that's it. As the disease outbreak, and another one important thing is the disease outbreak, and you are aware that the bacterial, viral diseases, and the protozoan and parasitic and innumerable diseases are there. Now, more recent research indicated that. Next slide. More recent research indicated that in the earlier days, and what we did was microscopic diagnosis, histological diagnosis, and there's a microbiological diagnosis. And immunological and molecular biology diagnosis is gaining momentous importance as far as diagnosis of the disease is concerned. Next slide. Look at the immunological diagnosis is a large precipitation, accumulation test, ELISA and dot ELISA, latex accumulation test, and also the fluorescent antibody test. And these are the more recent tests to find out the authentication of the species. These are pathogenic microorganisms which are affecting the host system. Next slide. Diagnosis, various molecular PCR techniques, and also the labeling and reduction of nucleic acid, resistance and digestion, and RFLP, AFLP, RAPD, and then DNA microarray. And more recently, and these are all the advanced techniques which are widely employed for the disease diagnosis in aquaculture operations. Okay. And I already told you, this is a probiotic microorganism playing an important role in the immune modulation, and how these are accelerating the phagocytic activities in the upper panel. And you might have, these are all the various immunological parameters being accelerated by the phagocytic activity, total hemocyte count, phenol oxidase enzyme, superoxidase anion, and the lysozyme. And these are all the systems. These are all the parameters which is being accelerated with the help of this probiotic microorganism. Okay. How then this product is nothing but this probiotic product. This is being synthesized by the probiotic microorganisms. When it's a phagocytic cell, that's a phagocytic cell will perform the functions of phagocytic activities. When the phagocytic cell is activated, this is an activated phagocytic cell. That's a phagocytic cell will do the phagocytic functions, and this product, if it will accelerated, it will capable of producing cytokines, various forms of cytokines, and when this produces the cytokines, and it is responsible for faster activation of the phagocytic cell, the phagocytic activity takes place at a faster rate, and this product is being extracted from the yeast cells, the probiotics, yeast probiotics. Next slide. These are immunological parameters, and this how these probiotic microbes are playing an important role in immunological parameters, non-specific immune responses, lysozyme content, phenol oxidase activity, superoxide dismutase, and as well as the bactericidal activities. And we are adding these probiotic microbes around the field that will accelerate the non-specific immune parameters. So this is a research which we have obtained from our own laboratory. Thank you. And another one techniques, more recent techniques which we are adapting in the hatchery environment is a bioencapsulation. Bioencapsulation of these probiotic microbes. What is bioencapsulation? As a aquaculturist, it's not as a bioencapsulation. Bioencapsulation means live feed organism may enrich with the certain material. That is called the bioenrichment. You are enriching the biological organism with the beneficial product. That is called bioenrichment. This same bioenrichment techniques or bioencapsulation techniques is also applicable for these probiotic microorganisms. By using the rotifer, rotifer is the live feed, and rotifer is encapsulated with the lactic acid bacteria. When rotifer alone, it will have its own efficiency in that aquaculture system. And when it is encapsulated with the specific probiotic microbes, and it will perform a better function. So bioencapsulation techniques by using the probiotic microbes. Okay. So Pages, bacterial Pages. You know what are Pages? What are Pages? Pages are the virus that will affect the bacteria. Okay. This same concept, this concept is being utilized in the aquaculture operations, and these bacterial Pages are only used as the probiotic microbes to use or to combat this pathogenic bacterial in that aquaculture environment. This is a bacterial Page techniques. Okay. Then the seaweed polysaccharides. Seaweed polysaccharides are only used as the immune modulator. Seaweed polysaccharide is the immune modulator. Apart from this, so far what I have narrated is these probiotic techniques, how probiotic techniques are used. The seaweed polysaccharide is only used as the immune modulator. Only used as the immune modulator. Next slide. It's antiviral activities. It's antiviral activities, antibacterial activities, and the immune modulation. There's three different activities are performing by the seaweed polysaccharides. And as far as antiviral activities is concerned, now this majority of the seaweed polysaccharides are sulfated seaweed polysaccharides. Polysaccharides, whatever the polysaccharides we are obtaining from the seaweeds, we got sodium alginate, alginic acid is the polysaccharides, and in majority of the polysaccharides we contain the sulfate as a group. The sulfate is a positively charged molecule. Sulfate is having positively charged molecule, and you are all aware that in the virus, when it is entering into the host system, and with the viral ethers, there is a load which is called the B3 load, and the B3 load is having amino acids, and those amino acids are the negatively charged molecules. And these negatively charged molecules will very much helpful. Negatively charged amino acid molecules which are present in the B3 lobe of the viral envelope is highly responsible for the attachment in the host system and the primary attachment and also the secondary attachment will be invaded. And what will happen when we are using this seaweed polysaccharide? Charged sulfate molecules present in the seaweed molecule. That will go and attach to the negatively charged amino acids which are present in the B3 lobe, and it will void and it will block the B3 lobe. Positive and negative charged molecule will block the B3 molecule. Ultimately, what will happen? This virus molecule, whatever the virus, they cannot be able to enter or include into the organism, and it will be eliminated. And by using these specific characteristic features, and now scientists all over the world, and they are trying to characterize the seaweed polysaccharides to combat the viral disease outbreak. Okay, next slide. This is immunomodulation, how these immunomodulatory activities take place, and here immune modulation. As far as immune modulation is concerned, and in majority of the aquatic organisms, first in the pyramid stream, and this pyramid stream is having lipopolysaccharide beta-glucan binding protein. Lipopolysaccharide beta-glucan binding site, and this specific binding site is having more than 3 motifs: glucose motifs, polysaccharide recognition motifs, and also the lamprey binding motif. And these motifs combined with seaweed polysaccharide, they will form the mixture of this polysaccharides, laminarin, and also the beta, that is beta glucan binding protein. And this admixture, what will happen? This admixture having the characteristic features of enhancing the activities of the propional oxidase enzyme system. When the propional oxidase enzyme system is enhanced, obviously we can get more resistant. The immunological parameter is automatically raised more than the required level, and it will give more resistance against the disease outbreak. And therefore, by using the antiviral properties and immune modulation properties, scientists all over the world, they are concentrating on characterization of seaweed polysaccharides as immune modulator as well as the antiviral molecules. Next slide. And we have also characterized this seaweed polysaccharide, picodan, alginic acid, sodium alginate in our laboratory. Next slide. And what we found out was this here, the boycott syndrome virus. There is called challenge test. By using the challenge test, we injected this boycott syndrome virus into the particular culture system. And that system was fed with the seaweed polysaccharides or different polysaccharides, the picodan, sodium alginate, and alginic acid. And we want to find out, just now I explained the theory, how these antiviral properties are there for the seaweed polysaccharide. How it is binding with the N3 lobe of the viral molecule envelope and with the positively charged molecules of seaweed sulfate present in the seaweed. And here we proved that this topping number, when the concentration of seaweed polysaccharides is enhancing, this DNA copying number of WSSB is reducing much in the sense, and it is controlling the colonization and also further proliferation of the virus molecules inside the host system. And this RT-PCR analysis, next slide. And we also found out there's an immunological parameters, immune gene expression. When the concentration of seaweed polysaccharides is increasing, the propional oxidase enzyme system, the gene responsible for enhancing the propional oxidase activities is also enhanced. Therefore, it indicated that and the seaweed polysaccharides are only used as the immune modulator and the antiviral agent. This is also one of the more recent research and which is going on as far as aquaculture is concerned. And the effluent treatment, as a zoologist is much aware that and what is the major constituent in the effluent, nitrogen and phosphate and phosphorus. What is the source of phosphorus in the aquaculture? This is general knowledge question. What is the source of phosphorus? Phosphorus accumulation is also high. What is the source of phosphorus? Accumulation of phosphorus in the aquaculture sediment. Anyone? What is the major component in the aquaculture field? Zeolite, the usage, everything has been done. Now everybody is looking for organic and that's a green aquaculture. In the aquaculture field contains more than 60% of the fish meal. Fish meal is being grinded where we are born. Fish bone, small fish bone is also being grinded. That contains phosphorus. And accumulation of phosphorus and the nitrogen and the phosphate, and these are all the major constituents as far as aquaculture effluent is concerned. Now, in olden days, as rightly pointed out to Sir, and geolite is only used to reduce the organic load in the aquaculture farm. Uses of geolite in the long term, and it will not yield any sustainability, and we cannot culture this organism for more than 3 or 4 cycles. Therefore, now uses of zeolite is completely avoided. Now, soil probiotics with the help of phosphate solubilizing bacteria, nitrogen oxidizing bacteria, and also the phosphorus solubilizing bacteria. Poly mixture of all these bacterial culture, and now it is only available in the commercial market, so as to take care of the accumulation of unwanted materials and excess nutrient materials which are accumulating in the aquaculture system. Okay. And the RAS, this is a more recent techniques. Because as far as this other countries concerned, in my first slide I told you we are having enormous amount of rivers, reservoirs, lakes, and lagoons, pools, and everything. But in other countries, and where they are very much constrained about the availability of the aquaculture land, and they are venturing into the RAS. It's a recirculating aquaculture system. As far as RAS is concerned, next slide. ROS. What is ROS? ROS is a recirculating aquaculture system. This recirculating aquaculture system is a component within the confined environment, very small area, high stocking density, more than 150 to 500 individual per square meter. Just imagine that. It is just like this crumbling or dumping. It's a very high concentration of people inside the small room. And in such environment, this ROS, and they are getting the maximum profit. How they are attaining the maximum profit? Here, next slide. Carbon dioxide removal is there. This here, this a diffusion, ultraviolet radiation, and ozone. This is the disinfectants of water quality parameter. Take care of the water quality parameter. I already told you accumulation of nitrogen is a biological filter, the fluidized bed filter, mixed bed filter. Various types of filters are there. Here the solid waste removal, and then also agitation of this aeration and agitation is also very high there. And then removal of dissolved solid and other organic removal. Here, this very small system, it is highly sophisticated system. As far as cost is concerned, very high. But within the limited area, the production is very high. It's more recently in majority of the countries which are involving into the venturing into the aquaculture system, they are adapting the ROS for getting the maximum production. This highly sophisticated system, the cost effectiveness. Next slide. Here, reduced water requirement, reduced area, very small area is required. Then the control of temperature, water, everything will be taken care of. And this in this ROS system. And instead of doing aquaculture operation in the hectare and hectare, 5 hectare, 2 hectare, or 10 hectare, and within the limited area of 1 cent or 2 cents and area, and they will produce more amount of fishes or more production, and with the help of the sophisticated instruments. It will take care of whatever may be the attachment, it will take care of. It won't deviate the specific water quality parameter or feed requirement, and everything will be met out by this highly sophisticated system. This is a ROS, it's a more recent aquaculture techniques. And next line. And another one is aquaponics. What is aquaponics? What is aquaponics? Yeah, simultaneously culture of very good, simultaneously culture of aquatic organisms and plants. So the plants which require less amount of soil. That is called the aquaponics. Aquaponics is also the more recent aquaculture techniques. And here the main concept is, this major excretion is ammonia. Ammonia is converted into nitrate and then nitrite, and nitrate is the fertilizer for the plant. And the plant materials will also take care of filtration of other unwanted nutrients which are available in the culture system. Therefore, we will get simultaneously more plant growth and more plant production, and also the aquaculture production. Such a type of techniques is called aquaponics. Aquaponics is also the more recent techniques which is widely adapted in the developed countries and also the developing countries, and to get more aquaculture production and for sustainability of aquaculture. Next slide. Another one important, next slide. And the bioblock technology. What is bioblock technology? Bioblock technology. This is a more recent technology as far as aquaculture is concerned. Carbon nitrogen, and this bioblock is nothing but the process. What is bioblock? Bioblocks are the aggregates of algae, bacteria, protozoans, and other particulate organic matter. That is called bioblock. And this bioblock is being developed with the help of addition of more amount of carbohydrate material into the culture system with vigorous aeration. You have to give vigorous aeration. When the vigorous aeration and also the carbohydrate materials, and it will facilitate the growth of the microalgae, bacteria, protozoans, and other particulate organic matter. And altogether, this aggregate is called, technically we are calling it as the bioblock. Okay. Now, when we are using this bioblock technology, what is the advantage? If you see the advantage, and the major advantage next slide. These are various things how this bioblock technology is performing. Go to the next slide. Okay, two critical service as far as this bioblock is concerned. One is treating waste inside the system itself. Okay, this bioblock, once the bioblock is formed, and they will take care of whatever may be the waste material which is accumulating into the aquaculture system. And second one is so providing nutrition to the aquatic organisms. Here, bacteria, RJ, protozoans, and other organic particulate matter, and they are highly essential for the aquatic organism for their own feed. Here, the feed is a natural food material in this system. And one hand, and we are treating the waste material within the system itself. And second one is whatever may be the bioblock formed due to this process, and they will act as the nutrient for those aquaculture organisms. And you need not spend much amount on the feed materials. It is almost feed on the nilla. If the system is perfect and the bioblocks are developed well, and you need not worry about the feed quality also. Here the major advantages is the minimal water exchange. What is the minimal water exchange? For example, 0.1 to 0.1 to 1%, 0.5 to 1%. You just imagine that whatever may be the aquaculture system, in the extensive aquaculture system, we need not worry about the water quality exchange. In the semi-intensive aquaculture system, 20% water exchange must be there. In the intensive culture system, nearly about 100%, 60 to 70% or 80% water exchange is there. In super intensive culture system, 200% or 300% water exchange must be there. But here, when we are introducing the bioblock technology into the aquaculture system, and two things we are getting benefit. There is only water, minimal water, 0.5 to 1% water exchange is enough to maintain the culture system in a perfect way and in a sustainable manner. And just benefit we are getting out of this bioblock technology. And when the minimal exchange is so as to enable this bioblock to develop better, and it's perform its function into the particular environment. That is a minimal water exchange. Then next slide. Another one important techniques I want to introduce is the notobiotic. Notobiotics. First one probiotics, then prebiotics, then probiotics and prebiotics mixed together one time. I forgot to spell out to use the symbiotic. When you use probiotic alone, it is called a probiotic technique. When you use prebiotic alone, you can use as a prebiotic. When you combine this probiotic and prebiotic to enhance the efficiency of probiotic, such technique is called a symbiotics. Okay. Now we have also studied something about the immune modulator, how these immune modulator as well as the antiviral compounds are there, and then ROS, and also the bioblock technology. Now here another one important technology. More recently, there's a people are doing as far as aquaculture research is concerned. It's a nanotechnology. That's a there's a notobiotics. What is notobiotics? Notobiotics. Again, everybody know what are notobiotics? Notobiotics in the sense organisms which are free from any microbes. That is called the notobiotics. Okay, notobiotics are microorganisms free from microbes. Number notobiota. Any organism notobiota organism illa ella organismthula vandu microbes irukku. Whatever the research we are doing, and we are taking selecting the particular fishes, and we are doing experimentation on testing the efficiency of the diet, and testing the hazardous effect of these particular chemicals, and all this we are doing in the non-notobiotic organisms. Therefore, whatever the microorganisms which are present on the surface and also the inside this host system, that will also show some resistance, and that will also have its own influence on the parameters what we are studying. And whatever the results, and we are believing that ignoring this microorganism influence, and we are believing that we are getting 100% result. Am I correct? Yes. If you want to get the authenticated scientific research, you have to do the research on notobiotic organisms. Notobiotic organism in the sense the organism which is hatched out in the microbial free environment is called notobiont. It won't have any microbes inside the system. But after hatching, you intentionally add some microbes, and into the system, and those microbes will enter into the gastrointestinal environment. And now you are aware that, and what are the microbes which are present inside this organism, and accordingly you can do the research. Okay, now that therefore notobiotic research will help the characterize the whatever may be the chemical, hazardous chemical, to test the efficacy of the feed, and you test this whatever may be the pharmaceutical product. Now, testing of pharmaceutical and efficiency, all feed efficiency is being done in the notobiotic organism. And whatever the results you are getting out of these notobiotic organisms, that is 100% result. And in the sense, and you are eliminating the influence of this microorganism or microbiota inside the gastrointestinal environment, and you are getting the result. And such type of research is also going on there. But one of the main tedious setback here is the production of notobionts. Production and maintenance of notobionts is very, very difficult, and yet research is also going on. Okay, in our laboratory also, we have studied some notobiotic research. This aquatic organisms are laying egg, fish egg later. Now, researchers found out that the microbes which are colonized in the surface of the egg of the egg of an organism, that will determine the hatchery fate of these young ones. That will determine the fate of the young ones. Whether it has to be hatched fully, or it has to be hatched in the abnormal condition, or it has to be hatched out in a full manner. Majority of the pathogenic microorganisms are invaded and they are occupied on the surface of the egg, in the sense that the deformities in the larvae may occur. And we developed certain technology with a combination of various antibiotics at various levels, and we sterilized the surface of the egg and make them to hatch out. The hatching percentage was very, very less, nearly about 20% when compared to that of the normal hatchability. And also we found out that deformities in these hatched young ones. Therefore, the microbes which are colonized in the surface of the egg playing an important role. In the similar way, microbes which are colonizing in the gastrointestinal environment and the surface of the organism playing an important role in testing its, in expressing its own efficiency and in testing whatever the product you want to test. Therefore, the researchers are also concentrating in the production of notobionts and to test the efficiency of this notobiotic research. Next slide. It's a nanotechnology, and everybody is talking about nanotechnology, and it is very easy for us to synthesis of 3 nanoparticles. Okay, 3 nanoparticles, and as you are aware that when we are synthesizing the particle at a nano size, obviously the efficiency is very high. Clean synthesis of the nanoparticles by using the various types of seaweeds and the various types of plant materials and so on and so forth. We are synthesizing the nanoparticle. No doubt that the nanoparticle playing an efficient role in enhancing the efficiency of this organism and also the improving health of the organism. Okay. Now let's do the nanotechnology. Next slide. This is a nanovacine. Nanovacines. So nanovacines are produced in 2 concepts. One, these nanomaterials, whatever the nanomaterials, and they are functioning. They are functioning as the immune modulation as well as the adjuvants, as a nanovacines. When we are preparing the nanomolecules, the phyton based nanoparticles, the phyton based, the phytosan based nanoparticles, and also the various nanoparticles are synthesized, and they are performing in 2 different functions, as immune modulation as well as adjuvants. And this nanovacines are performing in the slow release of this particular material into the system, and therefore nanovacines are getting momentous importance as far as aquaculture research is concerned, because mass vaccination is possible with the help of this nanovacines. Therefore, it is gaining momentous importance as far as nanovacine is concerned. Okay. Next slide. Next slide. Next. Okay. Now, another I want to introduce another 2 more terms and before my concluding, some metabolomics. What is metabolomics? Metabolomics. One of the more recent research which is going on as far as aquaculture is concerned. Okay. When you are starting the particular organism, okay, you are starting the particular organism. So, நம்ம செய்யற research தான், but இந்த செய்யறாங்க அவ்வளவுதான். You are starting the organism and you are taking the protein level, lipid level, and also the amino acids level, Artisa profile, and the histology, how the gills and other kidney and other organisms are damaged. And this is a basic research. And now the researchers are found out that when the organisms are subjected to some adverse condition, there are some metabolic there are some initiation in the metabolic activities, and that way there is a new metabolic pathways are there. And as the results, what will happen? Accumulation of certain metabolites are there. And if you are able to pinpoint this metabolite accumulation, and you can easily find out and what is the impact on this organism. And you are starving the organism, and you are taking the metabolites. What are the metabolites? By using highly sophisticated instrument, various metabolic end product can be easily characterized. And if the accumulation of particular metabolic intermediates are very high in the starving organism, in the sense that that particular metabolism is hampered, and due to that, these intermediates are accumulated at a higher level. Such study is called metabolomics. By studying the metabolic intermediates, and we can easily assess the impact of these particular food materials or particular hazardous materials or any impact on the organism. And with the help of this, and we can easily find out the what is actually happening there. Okay, you are offering the highly nutritious diet. After offering the nutritious diet, and you are taking the intermediate metabolic. Okay, intermediates, and in the sense, and with the help of this intermediate, you need not worry about anything. You go and have a subsample, and you do the in-depth analysis. You found out what is the intermediates are very high, and with the help of this intermediate, without disturbing entire population, you can find out that there is something is wrong into the health system. And with the help of this, and you can easily find out there is a metabolic resource. And next one. Next one, this is the various stages involved in metabolic research. Next one. There's a nutrigenomics. It is also one of the important research as far as aquaculture is concerned. Nutrigenomics. Whatever the nutrition which we are adding into the diet, it is digested and assimilated. And not only it is assimilated, and it is triggering its genome, gene expression, transcription, protein synthesis, and also it will accelerate the metabolomics. There is a microbolomics. Microbolomics in the sense genes related to the particular microbolomics and microbiota. And these are all the things which are happening inside the living system when we are adding the nutrition in the diet. And we are stopping our research in the sense of taking the growth and taking the protein concentration of protein, lipid, and other things. And we fail to do some in-depth research in this area. And if you go for the in-depth research, and you can do some wonderful research as far as nutrigenomics is concerned. And with the help of this, after adding the nutrition, you can go for the expression of particular gene, and how the protein, what is the expression of particular protein, and then the proteomics, and also the micromics. Micromics is a genes responsible for microorganism and microbiota. This is a proteomics. Next slide. This is my last slide related to the more recent advantages. This is climate change. Climate change is one of the hottest subject as far as our any area is concerned. Now research is also going on. This climate change resistant organisms and microalgal production and heat for sea urchin formation and open sea aquaculture. And these are all some more recent research which is going on as far as aquaculture is concerned. Thank you. I will be there for another half an hour to 1 hour here, and if you have any doubt, and we will have the interaction because of the lack of time. And I think with the permission of these organizers, shall we close this session? Raja?
Okay, sir. Yes, sir.
Yeah.
Thank you, sir, for addressing the issues, challenges, and the status of aquaculture in India and the significance of probiotics, seaweed polysaccharides, grass, aquaponics, and biofloc technology in enhancing the desirable characteristics as well as sustainability of aquaculture in India. Thank you once again, sir, on behalf of the organizing committee. Now we shall part for tea break, and we will gather again at 12:05. The tea is available outside. Thank you.
அப்படியே இதே ஸ்கிரீன்ல
சப்ஜெக்ட் கொடுப்போம் அதோட சேர்த்து ஓடி வந்து
of resource is going very rapidly. Meanwhile, it is our duty to, we want to produce lot of species or lot of biomass of quantity of aquatic organisms it must, because of to eliminate or eradicate malnutrition, or we want to enrich nutritional support through the aquaculture system. So that is the reason I have given here food security. So how even you knows aquaculture can contribute as per this title, but what way the aquaponics will contribute? So that is the question mark. Even our professor Parvez are already pointed out. There are a lot of evaluation in the field of aquaculture. Is that what is the aquaponics? What is our contributions? So what are the gaps are there? How we can do the practices in better way? Please, next slide. Yeah, you are so much here. Now, aquaculture is a very happy and beautiful culture. There are a lot of beautiful fishes are there, like you. So you should have yourself. So what is the aquaponics? Already I know, sir. Sir came along with Professor Haripa sir to my college. I think 1 year back. Yes. What is the aquaponics? Why it's needed? Yes, it's needed because of. So we have huge potential, but that potential, whether it's readily accessible or available, that is the question mark. Because of always the pollution also interfering in our life. Due to the pollution, there are a lot of issues. I don't want to explain the things. So even though even the pollution problem also, there is no solution. Because of it's a continue enrichment of nutrients from our waste or natural waste, whatever it is. So we want to adapt the new technology. Yeah, because of we want to food. Without food, there is no possibility of expand of human population, not only human population, all the other populations. Now, recent study shows that now the human population becomes standing or going to decline. So that you should understand. So in that way, the aquaponic system is very important. What is the special characters in aquaponic system? I think you will be knows about the agriculture system. What is that agriculture? Agriculture means plant we can cultivate. What is the substrate? Soil. But here the soil we can replace soil-like material like cocoa. So compared to the agriculture, we have the agriculture family. Everyone says that nearly 70% of rural population depending on agriculture. The total our economy contributed by the agriculture product, but the same things we want to improve. The culture system means the main reason we want to produce anything within the short period and high biomass. Even that concept. So this aquaponic system, we can produce two different protein source. One is if you are vegetarian, you can get vegetables. If you are non-veg, then you can get non-veg. The thing is, water is safe. The water is safe. So here, how we are consuming the water, how we are improving our production system. That is the challengeable task for us. This is not yet challengeable. That I will tell you later. Next. So already we discussed the same things. This is the water efficient system. If suppose if you are using 100 liters in this system, only the absorption are utilizing only 10%. That means 10 liter. Rest of the 90 liter of water we can store. The same things if you are using the open environment, what it will be happened? Either evaporation or percolation. One மேல போவோம் இல்ல கீழ போவோம். வாட்டர் அப்போ ஒன்னு ஆவிய போவோம் இல்ல பூமியில கிரவுண்டுக்குள்ள போவோம். அப்போ இந்த இஷ்யூஸ் வந்து நியர்லி 40% அந்த இஷ்யூஸ் இருக்கு. அப்போ நம்மளால இட்ஸ் வெரி டிஃபிகல்ட் டு ஆல்வேஸ் getting more water. So our forefather also says that what is that நீரின்றி அமையாது. There is no life without the water. So the water is unique. There is no technology to produce water up to date. There is no technology. So still we want to defending the natural phenomena. So in this aspect, so even whatever the human being not able to perform, even we can save the water or the minimum water, we can produce large quantity of food materials. Large quantity of food materials. So nowadays everyone is speaking about organic. Yes, organic means so there are a lot of controversy. But in the aquaculture system, in our experience, I think we can produce in the organic manner. That experimental setup and the results also I will tell you later. Next, sir. So another thing, compared to even the bore water, what is the problem? You can irrigate, but additionally you want to support with fertilizer. But aquaponics, no need to add any fertilizers. Because of the fertilizer itself is as a nutrient, it's contained in the fish water. So in that aspect, there are 10 essential nutrients which are very important for plant growth. So the same things, even another thing, the process of mineralization is very fast compared to soil. These are all the advantages while you are using the aquaponics system. Bore அடிக்குதா? Bore அடிச்சா சொல்லுங்க, ஆஃப் பண்ணிக்கலாம். தண்ணிக்கு ரொம்ப bore அடிக்குது. சரி, anyway, good. Just what I want to know, we want to interact more, because of we are the scientific forum. There is no mistake. So always I am expecting from your side more queries or questions. Next, sir. Is that next one? Okay, next. Okay, this is the one of the important story. Just I want to convey to you. Actually, you could see the picture. So, how that people are working in the aquaponics system. So, as per the reports, what it says, the first RGI, or documented of aquaponics system, where in the Mexico. I have pointed out that. So nearest, especially the Central America area. Another one important point, the name also they have given like Aztec Indian. That means Narwhal Indian. Angeka Indian will go on. That is the question mark. Okay, anyway, I will come to your point. So about the same things, another thing, the near to the lake, there is the place. What is the name? Do you know? Chittalan. Chittalan means it's a South Indian name. How that name also is goes in Central America. So that means we are the pioneer, we are the technology developer. This technology originated from us. The thing is, the report maybe, the documentation maybe in the Central America. The dress code also is look like our ancestor of Indians. So another thing, so another point I mentioned here, even they have documented in the 16th century. But before that, nearly 15th century, we already practicing one culture technology in Kerala. How many are from Kerala? Okay. The technology is called Tokali. Pisho, plant, the same concept. That means நம்ம யூசுவலா இன்னைக்கு நிறைய போட்டோ எடுக்க சொல்றேன் சார். ஏன்னா புதுசா இப்ப பேச வரும்போது நமக்கு டாக்குமெண்ட் வேணும். ரெகுலரா இதே வேலையா இருந்தா நான் சொல்லணும் அப்ப சொல்ல மாட்டோம். ஸோ அந்த மாதிரி that means அவங்க some of the documents are the foreign countries they have done in good manner, but these are routine activity. It may be a reason we are not documented properly. Next. So see, I think these part all are very interesting, very women's very active women's. So the women's role is very high compared to the men's. Because of women's always liking the they want to produce. Even another things I want to find out, who of agriculture, women's. It's still there is a practices like the களை எடுக்கிறது, நெல்லு நடுறது, நாத்து நடுறது, விமென்ஸ். ஸோ அவங்க தான் அதை பண்ணியிருக்காங்க. ஸோ அப்போ அதே மாதிரி அந்த பியூட்டிஃபுல், அந்த பியூட்டி பண்றது அந்த அதெல்லாம் யார் பண்ணுவா? விமென்ஸ் பண்ணுவா. ஏன் ஜென்ஸ் பண்ண மாட்டாங்க? அவங்க நேச்சுரல் பியூட்டி. அப்போ இது ஜோக் இல்ல, நீங்க மயில்ல இருந்து சேவல் வரைக்கும் பாரு, இங்க கூட பாரு. சரி, அப்போ innovative system. The innovative system. Mainly, the women are involving very more. This is the MPDA, Ministry of Commerce, Export Development Authority. They are providing fund. If you interested, you can produce the food. ஏன்னா previously நம்ம என்ன பண்ணோம், காட்டுக்கு போகணும், நைட்ல தான் என்ன போச்சோம், தண்ணி பாய்ச்சோம், ஏன்னா எவாபரேஷன் கம்மியா இருக்கும், அப்போ பாம்பு கிடைக்கும், நிறைய பிரச்சனை வரும். இப்ப அப்படி இல்ல, காடை எங்க கொண்டு வந்துடும், வீட்டுக்கு கொண்டு வந்துடும். The same system we can do in our front area and a backyard area or terrace in the garden. So likewise, the women's Karnataka and Kerala, so many places the women are involving to adapt these practices. Next, not only women, see, don't worry, I am also a man. Sorry. Next, see how the same women, how she is adapted the same technology. Men, how he is doing. So whatever waste pipes or whatever things we have in our home, that we can do easily. Another thing, this is also aquaponics system. Even if you want to purchase 1 acre land, is very difficult nowadays. You might be knows the same things. இங்க வேணா தமிழ் பண்ணி தண்ணி இருக்கலாம், பட் நிறைய ஊர்ல தண்ணி கிடையாது, அப்படிதானே? அப்போ அந்த தண்ணியில உங்களுக்கு this system is maybe very fit because of this is the highly expensive system. அப்ப aquaponic நீ சிம்பிளா பண்ணலாம், எக்ஸ்பென்சிவா பண்ணலாம். அப்ப எக்ஸ்பென்சிவா பண்ணும்போது there will be more cost. Another thing, you want to some sensor based sophisticated systems. So you are educated, geology background, then you can do this kind of work. But the thing of even whatever production system, இப்போ ஒரு vegetable 10 ரூபாய்க்கு கத்திரிக்காய் வாங்குறேன்னு வச்சுக்கோங்க, அது 10 லட்சம் போட்டு அதுக்கு டிசைன் பண்ண முடியாது. அப்ப என்ன, what you are going to produce, according to that you can simplify the system. This is the basic things. இப்போ கஞ்சா வளர்க்கணும்னா it's okay, because of that is the more costly compared to vegetables. Next, another, see how that even the one pipe, how the peoples are the food flowering plants they are culturing. Next, another things, vendica, not only this, next slide, so many vegetables we can cultivate. Already the people are doing in foreign countries like Israel and other countries. Why our Indian people not that much interested to adopt this kind of technology? The reason, we have plenty of resource. So now it's okay. In future, definitely I will say this technology will emerge in our country also. Next, see, so not only so water, many of crop patterns, fish and vegetables, they go succeed produced and standardized. Next, so these are the suitable fishes, candidate fishes, aquaculture species we can use for aquaponics species. So what is meaning of candidate species? Which are the species easily adaptable for another, while producing the high growth and the biomass, the species may be a candidate species. So the same aspect, whatever species we are doing in aquaculture, the same species may be suitable for aquaponics. Next, even not only aquaculture, aquarium business today ரொம்ப எங்க பார்த்தாலும் எமர்ஜி ஆயிட்டு. எங்க காலேஜ்ல போனாங்க aquarium வச்சிருக்கோம். We have we are doing some of the breeding activity of aquariums. Likewise, the koi and the tropical fishes are highly adaptable in that. Another things, even now some of the issues it's come, if you are producing the aquarium, automatically that will generate more waste, organic waste. That waste, what you are doing? You are doing now we simply discard into the environment. That no needed. The same waste we can recycle it. The same waste we can use as a nutrient. the same waste we can produce plant-based proteins. So, those who are having our home aquarium are going to install aquarium facility. Similarly, we think our beauty is important. So, other than that, our food also is very important. So, you should think about that. These are the fishes highly adaptable. Next. See, one small tank, man, or light board. Just a key up green. Other snacks and the key up next. You are spending money. At the same time, you want to get more of them. So see how the people are so one side beauty, another side food. Next. Whatever you have thought, according to that, whatever money you have, according to that, you can even old material, the waste material also you can use it. Next. So mainly, what is the reason of advantage? Even there are a lot of even hydroponics are there. What is meaning of hydroponics? Without fish, if you are using the water, that is called hydroponics. If you are adding fish, then it is another aquaponics. So because of the nutrients we can get from fish waste. It's a simple logic. Next. So one of our students, she involved this aquaponic works, and we are also faced a lot of problems while any unknown technology or unstandardized technology or anything if you are doing, you are the beginner. So at that time, we are also faced a lot of problems, less growth, more disease, more efficiency. To solve that issues, my scholars intensively work. Then we solved that issues. How? So aquaponics, some of the nutrients like iron and 2 or 3 elements may be lacking in the aquaponics water, fish-based water. That same nutrients we want as a supplement. So where the nutrients are very high in the seaweed. So that is the reason we used seaweed as one of the components as a supplementary, maybe a biofertilizer. Next. Even after when we are used the same seaweeds for then scientific name next. The same we used liquid fertilizers. Next. Already I told you some iron deficiency we can compensate. Next. So this is our model. So this is simple thanni or another aquatic system along with fish with another nutrient cycles. Thanni mele and another plant based. So double layer system. That's our model just we showed here. Next, sir. So this is our student. She done in her home. Don't want to spend much instrument or money. Simple. Just this is enough for you for the experimental purpose. Another thing, aquaponics we can categorize raft, net, nutrient film technology. Raft means floating technology. Another one, media filter technology. So which technology you are able to adapt, that you can do, no issues. That on our over technology, fish, plant suitable. Next. So one of my student, MSc student, she done algae, spirulina, along with aquaponic water. We cultured onion plant. So in this plant, when we are using the aquaponics waters, we are able to get chlorophyll, high chlorophyll content. So that which plant having high chlorophyll, it shows that healthy of the plant. So in that aspect, she done wonderful work. Even some of the people, you may criticize, research in our onion she done this work. Anyway, next. So this is the algal. Easier to பண்ணிருக்கோம் ஸ்பைரல் இதுல என்ன ஒரு இஷ்யூ வரும்னா அப்ப நினைக்கலாம் சோ algae is the highly nutrient third world warல சரி second world war third warல வரும் second world warல பாத்தீங்கன்னா ரொம்ப ஒரு இம்பார்ட்டன்ட் ஒரு புரோட்டீன் சோர்ஸ் ஆனது ஸ்பைரலினா ப்ளூகின் algae இப்போ இதே பார்த்தோம்னா இது இதே நாங்க அக்வாபோனிக் வாட்டர்ல வளர்க்கக்கூடாதுன்னு ட்ரை பண்றோம் வளர்த்தோம் ஆனா இதுல என்ன இஷ்யூ வருதுன்னா ஆல்ரெடி அக்வாபோனிக் வாட்டர் வந்து maybe contaminated. Because of already our Professor Pallavisan pointed out, there are lot of microbes may be present in the feed and the gut content. So the same contaminations may be where may be present in the algae. So we can't say it may be a good quality algae, but if suppose if you want to process the same algae, 1 kg may be 1,200. The same algae we can we don't want to go for pharmaceutical grade. Yeah, why can't we go for a foreign grade? நம்ம தீவனத்துக்கு பயன்படுத்தக்கூடாது என்ன ப்ராப்ளம்ஸ் இருக்கோ அதுக்கு ஏற்ற மாதிரி நம்ம சொல்யூஷனை மாற்றிக்கிறோம் அப்புறம் some of the methods some of my students are doing another killing of microbes and removal of contamination from the algae. So that kind of work also we can do. Next. So this is the system which we have in our college. Next. So the same copepith is a very important component. That is the replacement using replacement of soil. And another thing, soil வந்து என்ன பண்ணுவோம் வேற ரூட் போறது கொஞ்சம் சிரமப்படும் ஆனா இந்த copepith வந்து சாஃப்டா இருக்கும் ஸ்பாஞ்ச் இருக்கும் நீங்க just whatever waters you are pouring along with nutrients automatically it will be stable. So the root system also easily can penetrate in the copepith. So our available nutrient also maybe wholly very more compared to soils. Another thing is if you are using the soil, then the microbial work we need very high because of the mineralization process is very important. But aquaculture is not like that. So already mineralized or active nutrients are here. This is the more advantage while you are using aquaculture. Next. So what are the tanks? How many liters of water they used? Everything we have given here. Next, sir. Time next. So this is the plants. Another. One of the kali solarium. Nothing. Kotamali there. Coradium. Abranada. Trigonella. Many plants we attempted in our research. So we got very good result. So this is the eye open for you. It is going to be a good work for your project work. So already I have given within short period we can complete it. If you are choosing other research problem, it will take more time. So already I have given stock in 10 different colleges. All colleges they have started their own aquaponic system in their college. So likewise, what is my request? Sir is here and the professor also is here. So you may take some initiative, cost effective, no need too much of money. So anyway, you think about it. Next. Already different type of seaweeds we used. So how for the efficiency? Why you are using different seaweeds? That was we carried out in our lab. Next. So it's a general parameters, whatever compositions are present in the seaweeds. Next. See how that replacement the whatever nutrients lacking in the water, aquaponic water, the same how we can compensate. So even that another thing see here iron level that is the more important manganese then other copper and zinc. So these are all the micronutrients maybe may not available in the aquaponic that we can replace by using of these plants. Next. Next. Yes, sir. Wait பண்ணுமா இல்ல நான் கண்டினியூ பண்ணலாமா? Okay. Next. Same. அப்புறம் already சார் சொன்னாரு என்னது பலவேச சார் if suppose any system if you want to produce in an effective way we want to strengthen or we want to increase the growth even that aspect my PhD scholar she used some microbes probiotic microbes that microbes she got from the fish cut the same microbes she cultivated and again used in the water for mineralization purpose. Next. So based on we already deposited in the NCBA. So especially the Lactobacillus plantarum, that species she used. These species are effectively working for fertilization purpose and for production of more aquaponic. Next. So this is it's a general tables. Next. So we can say when you are using the aquaponics or along with these probiotics, what is the percentage? How far the fishes are happily growing in the system? So based on this result, we can understand the same seaweeds. Next. Here we mentioned what is the composition. Groundwater, cocoa with fish tank water, seaweed extract, cocoa with. So likewise, different composition lab, which composition may be best? We studied in our lab. Next. Along with tilapia. Tilapia also one of the important carrier fish in the world. The second largest biomass of productions in the tilapia. Worldwide. Next. Next. So we did the experiment 70 intervals. Next. See, this is the results we got. So different seeds, same species, we used a different concentration or a different composition of substrate. In that, see, healthy seeds are there. That means within the short period, we are able to grow very healthy and highest growth seeds while you are using the aquaponics. தமிழ்ல ஒரு பழமொழி சொல்லுவாங்க தெரியுமா? ஒரு பயிர் வந்து அதோட இளமையிலே தெரியும் எப்படி ஃபியூச்சர்ல இருக்கு. So அந்த மாதிரி so the aquaponics waterல we got good result. Next. So anyway, it's good, because of our forefathers, their insight is very entirely different for us. Next. So that's the same things. Other things here we use vermicompost. So everywhere the people talking about vermicompost. I am also opposite. The thing is, if you are cultivating any plant, over nutrient also that will give stress to the plant. Point one. Second point, when you are using the vermicompost, that mineralization process or active elements to the plant, it will take 10 to 1 week. But aquaponics is not like immediately you can utilize the plant can utilize. So glucose போறதுக்கும் சாப்பிடுறதுக்கு வித்தியாசம் இருக்குல்ல, அதுதான். Next. Same. Next. இதுல பாத்தீங்கன்னா so nitrate எவ்வளவு இருக்கு? Nitrate, phosphate, magnesium, calcium. So all the elements for purpose need for the plant growth, especially root formation, chlorophyll production, cell divisions. Next. Next, sir. So likewise other micronutrients, how far it's effectively working for the plant growth. Next. The same another things while you are using the seaweeds, it's contain lot of growth hormone. Another growth hormone, just you can spell out. Very good. I'm very happy because everyone are not sleeping. Next. 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 Okay. Next. This is the result that we got. Again, a picture comes already showed one picture. How far it has gone. The same results. Next. 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 Our system. Next. See what our student did. What is that? Which plant is it? என்னப்பா இங்க ரொம்ப டிஸ்டன்ஸ்ல இருக்கறதா தெரியல, நான் தப்பு சொல்றேன். மிளகாப்பா சில்லி, அது நான் சொல்லி தரேன், அதுதான் டீச்சர். அப்போ ஜஸ்ட் யூ see here அந்த லிக்விடிங் ஸ்டேஜ் ஆஃப் சில்லி, இது வந்து மேனிபுலேட் பண்ண முடியாது. ஓகே. நெக்ஸ்ட். How the growth even height weight of the plants. Next. Next, sir. Next. Next. Here a healthy, how we assessed the plant healthiness based on the chlorophyll content. Next. Another one more important point I want to add to you. Actually, we are doing all the things in the freshwater aquaculture system. The same system only worldwide they are doing. But now recent year, there are some techniques are emerging from marine and brackish water based aquaculture system. I will tell you later. Here, இப்போ ஒரு simple logic. Aquaculture நீங்க பயப்பட don't want to get after yourself because of if you take one lake, there are some plants. How it's grown. Inside of there is some lot of organisms. The same techniques. That is natural. The same techniques we are adapting in our laboratory. But there we can't assume the production level. Here we can assume the production level. There is the mineral or other components not in our hand. Everything depends on the environment. Here everything in our hand we can manage. So this is the simple things. Next. So even some of the countries they attempt some plants, even barley rice from the maritime water. Nowadays the scientists are working for genetically tolerant, saline tolerant plant species. So even that aspect, I think in future the aquaponic system will be flourished. Barley, many of the rice, see even some of the university they have done rice cultivation where in the ship கப்பல்ல என்ன பண்றாங்க நெல் விதை வைக்கிறாங்க இந்த கடல் தண்ணி யூஸ் பண்ணி அப்ப in futureல we can expect a more research output. It will publish that may be very useful for us. Next, brackish water system is a modern system how we can adapt in our home. Next, see the same brackish water we are holding lot of nearly 1,52,000 hectares in India. Everywhere the brackish water. So simply we are using only 1 gram like fish. Why can't we go for 2-layer crop? What the government of India insisting within the short period, you for one drop of water, how much gram of food you are producing? Now the technology is அன்னைக்கு மாதிரி ஓப்பனா வயல்ல தண்ணி விட்டு உட்கார்ந்துக்கிறது இன்னைக்கு டைம் இல்லை. So according to that, you just elevate yourself how we can adapt this system now. Home lab. Next. See already a salt tolerant plant. This is a naturally salt tolerant plant. This is the ideal plants for a brackish water environment. What is meaning of brackish water? உங்களுக்கு தெரியும் நினைக்கிறேன். Next. நான் already சொன்னல fish come. It's a traditional system. But according to our place, economic positions, just be modified. That's all. Next. See, in the year of 2014, where I am studying there, these are all my students, like your UG students. We organized. That is the person. He got failure. He is the consultant of aquaponics. Many of the places he installed in Coimbatore. What happened? This is the one roof now installed in developer. Already I am put to be considered. Sir, it's growing very well. Fruits also is coming. Ana kai வந்து கொட்டிருது. The fruits not going into the ripening stage. So this are his complaint. Then I said, okay sir, we will try. Then அவர் என்ன பண்ணாரு? The same system, his own system, he donated us without any money. So he is the best person. His support, we are able to construct own aquaponic system in our college. Then our students are intensively working. So I want to acknowledge him in this part, not only here, in this part very well. So just the second BSc zoology observed. Next slide we can. So this is our secretary madam, our college owner, Madam belonging to zoology, Madam father also zoology. So our college is top priority zoology. I think you might be know, sir. See, in our aquaponics, first என்ன வந்திருக்கு அந்த பூ பூத்திருக்கு ஃப்ளவரிங் வந்திருக்கு டொமேட்டோ உடனே மேடம் வந்து நான் கொண்டு போய் காமிச்சேன் ஸோ மேடம் ஓகே எனிவே யூ அட்டெண்ட் somewhat result but still we want to wait up to the fruits. Anyway, thank you. I said okay madam, next we go next. See, we are able to produce fruits. This is our success. Even compared to the natural tomato, It's a highly, taste, entirely different taste. There is no fertilizer. The coloration also we can understand how like it's look like good. Next, this professor Hanifa, along with the sir also came to know that he was college professor. So he's also visited there in the Palakira. Here like spinach and all. At the time of visit pannaga. Next, your minister DOD, Ministry of Ocean Development advisor. He's also came and many other people regularly they visited and all. Because it's a successful model. We have very good with whatever problems we solved it, and now we are planning to commercialize even some of the demand system. We did some diet with the industries for fund these industries. Some of the even this is the wrapped method. We don't want to spend more money. Just 1 week college ஒரு சின்ன இந்த மாதிரி போட்டுருந்தாங்க போரியோ அது வேற சிமெண்ட்ல போறோம்னு சொல்லி தூக்கி வெளியே போட்டாங்க. அது நான் எடுத்து வந்து என்ன பண்ணேன், அதை திருப்பி வச்சேன். அதுக்கு மேல என்ன பண்ணேன், பிளாஸ்டிக் அந்த தார்பாலின் போட்டேன், டேங்கர் மாத்திட்டேன். அப்ப வேஸ்ட் பிஸ்ல வேஸ்டையும் நம்ம என்ன பண்றோம், ப்ரொடக்டிவிட்டியா மாற்றோம். அவங்க தூக்கி போற அதே மாதிரி எங்க பசங்க எல்லாம் என்ன பண்ணுவாங்கன்னா இந்த பில்டிங் உடைக்கும் போது கிளாஸஸ் எல்லாம் கழட்டி வைப்பாங்க. அதை எடுத்துட்டு வந்து டேங்க் செய்வோம். எங்க லேப்ல எங்க ஸ்டூடெண்ட் செய்யறாங்க. அப்ப இந்த கிளாஸ் காணாம போனாலும் நேரம் எங்க அப்பர் தான் வருவாங்க. Next. So our student, she is working very intensively. We produced a fish that we gave to the women entrepreneurs, those who are in the nearest to the Coimbatore. Our secretary and along with that is the industrial person, one of my friends. We and with the diet, we produced some aqua tank cleaner products. That was also very interesting topic to us. Next. So this is the outcome. So Krishna Priya, these are all the people, second MSc, sorry, second BSc, Malini, UG students. Next, they have published many papers. Next, so even next. Okay. After these publications, I went to one national conference in Visakhapatnam. ISCA organized. There I presented my work. Even with the heavy competition, we able to win the best overall research award. This is the great output for us. Because of just this is not a magic. நம்ம வந்து இவ்வளோ ப்ரூவ் பண்ணி தான் நாங்கள் என்ன பண்றோம், ப்ரெசென்ட் பண்றோம். So அதனால the science farm also recognizing us. Even the same things I want to thank to Anagaraja sir and Madam and Raja sir given opportunity to interact with you. Now likewise wherever we go, so the same things if I educating to you. So what I am expecting, some of the students you should come with some adaptation or new technology. Next. So the same things I want to acknowledge my institution where I studied. I was studied at National College, Trichy. Then I studied at Annamalai University, Varayambalaji. Then I did my PhD in SIPA, Central Institute of Science, also worked with us. Yes, sir. Last 10 years I was the ICR, Ministry of Agriculture. Afterwards, I got posted to the CMFI, Central Marine Fisheries. These are all the by the education background institutions. With their moral support, still I am working in active mode. Another thing I want to, all the credit goes to our President, Dr. R.H. One of the greatest personality, zoology, and the Secretary of Wasik All India Private College Association President, as well as Indian Science Congress, one of the important persons. So now, 2 weeks before he was expected. So anyway, whatever it is, the same things also I want to acknowledge in this forum. So whatever achievements, everything goes through my students as well as my seniors, my colleagues. So if you have any doubts, we can interact. Otherwise, start session also is there. Madam, what I should do? Shall we? Please, any instructions?
Maybe 1 or 2, sir.
1 or 2. Please. Good. Okay,
Sir, I have few doubts.
first thing is நீங்க starting பண்ணும்போது
சொல்லுங்க.
ஸ்டார்ட் பண்ணும்போது low level செட்டப்ல பண்ண முடியும். இது வரைக்கும் across the world எந்த கம்பெனில ட்ரை பண்ணும்போதும் அந்த செட்டப் காஸ்ட் அதிகமா இருக்கு அப்படினா that's first question. செகண்ட் வந்து கமர்ஷியலைஸ் பண்றோம் அப்படிங்கல பட் இஸ்ரேல்லே கூட அவங்க கமர்ஷியலைஸ் பண்ண முடியாம now they are dropping it. And the FAO board இருந்து they are talking about developing something else because it's the work of dark lab. That's the second question. My third question is suppose if valences எல்லாம் okay and the valence crop வந்து some disease or some insect attack வருதுன்னா will we be able to add pesticide or insecticide such that the organism in the water doesn't get affected? That's my third question.
Yeah. Finished. Okay. Now, what people are going to do is first ஒரு question கேளுங்க ஆன்சர் சொல்றேன் அப்புறம் okay
Commercialization.
Yes, I can say it's not a magic, it's a kind of commercial vehicle. The reason why the people are giving poor response, they have alternative source. That is the reason we are not giving more important. Another thing, poor understanding of nutrient nutrient recirculation. No, FAO was highly appreciated and they have released manuals also. We have FAO based manuals. Even this country highly successful model for poor countries. Even those poor don't have the facilities, even they can do in their backyard. It's highly adaptable, successful. There is no doubt. Because of the thing is, still there is no SOP. What is meaning of SOP means? Standard operating procedure. That means that we can do. Then there is one issues like fish waste and plant growth. There is some relationship that we want to do a study. Because of hydroponic may be a failure because of we don't know how much of nutrients required by plants. But fish not like that. Even already I told you there are some lacking of ions like iron and other elements that also we can replace by using our supplementary.
Then you can propose the SOP to the FAO. If you are successful in your system means you can propose the SOP to the FAO.
Yeah, sure. In future we will do, sir. What is actually we are doing a small scale setup and experimental based. We are academic institution. If any agriculture based or any production unit, if anybody interested, then we can suggest, we can monitor, we can work together. If we got even நிறைய ரிசல்ட் இந்த ஆர்டிகல்ஸ் இருக்கு, நிறைய கேப்பேஜ் நிறைய எல்லாம் கண்டிப்பா பண்ணிட்டு இருக்காங்க. சரி, அது மாதிரி பண்ணலாம். ஆனா ஒவ்வொரு இப்ப பாத்தீங்கன்னா ஒவ்வொரு பிளான்ட்டுக்கும் ஒரு பிராக்டிஸ் சொல்ல முடியாது. ஏன்னா அங்க இருக்க நியூட்ரியன், ஃபார் எக்ஸாம்பிள் இங்க கிரவுண்ட் வாட்டர்ல நியூட்ரியன் வர மாதிரி இருக்கும். அதே குவான்டிட்டி ஆஃப் சப்ளிமெண்ட்ரி போய் நான் கோயம்புத்தூர் வாட்டர்ல போட முடியும். புரியுது? ஏன்னா கோயம்புத்தூர் வாட்டர் கிரவுண்ட் வாட்டருடைய நியூட்ரியன் ப்ரொஃபைல் வேற மாதிரி இருக்கும். தரவா நம்ம அண்டர்ஸ்டாண்ட் பண்ணி எல்லாத்தையும் மானிட்டரிங் பண்ணி கரெக்டான சப்ளிமெண்ட்ரி கொடுத்தா சக்சஸ்ஃபுல். என்ன நீங்க என்ன பண்ணலாம், இங்க இன்னைக்கு ஈவினிங் நாங்க அல்வா வாங்க போறேன், என்னால கோயம்புத்தூர் வாங்க முடியாதா? இந்த வாட்டருக்கு இந்த டேஸ்ட் இங்கதான் அவருக்கு கிடைக்கும். அப்ப அதுக்காக நீங்க வந்து திருநெல்வேலி அல்வாவே கேட்குன்னு சொல்ல முடியுமா? நீங்க பண்ண முடியாதுன்னு சொல்ல முடியுமா? என்ன கொஞ்சம் அண்டர்ஸ்டாண்ட், சப்ஜெக்ட் அண்டர்ஸ்டாண்ட் பண்ணுங்க. பண்ணிட்டு அதுக்கேற்ற மாதிரி எந்த பிளேஸ்க்கு என்ன மாதிரி நியூட்ரியன் ப்ரொஃபைல் இருக்கு கிரவுண்ட் வாட்டர். கோயம்புத்தூர்ல பாத்தீங்கன்னா அந்த ஆர்னமெண்டல் ஃபிஷ்ல சில வெட்டி பயங்கரமா வருது வித்தவுட் எனி நியூட்ரியன் யூசிங் த நியூ டெக்னாலஜி. ஆனா அதே ஃபிஷஸ் இங்க வரமாட்டேங்குது. எங்க திருநெல்வேலியில ஏன்? அங்க வாட்டர் வந்து ஹார்ட் வாட்டர், இங்க சாஃப்ட் வாட்டர். அப்போ ஹார்ட் வாட்டருக்கு என்ன ஃபிஷ் வரும், சாஃப்ட் வாட்டருக்கு என்ன ஃபிஷ் வரும்? வாட்டருக்கு எந்த ஃபிஷும் வராதுன்னு சொல்லக்கூடாது. Next.
Sir, before recommending any traits or the variety, it should be practiced or it should be acclimatized in various agro conditions, agro climatic conditions. If it is succeeded, then only it will be released. It's a usual procedure.
Thank you, sir. It's valid suggestions. The same things when the people working, some of the countries they able to produce successful crops. Even I will tell you, even they able to they mentioned like if 1 square meter we can produce 1.2 kg of fish. The same things we can produce nearly 600 to 1.2 gram of plant. அப்போ கீரைக்கு வேறையாகும். இந்த வாட்டர் டியூப் பிளான்ட்டுக்கு அதனுடைய இது வேறையாகும். எல்லா அக்வாபோனிக்ஸ்ல நீ எல்லாருடைய அந்த பிளான்ட் ஃபிஷ் காம்பினேஷன் இருக்கு. இப்போ கோல்டு ஃபிஷ் வைக்கிறீங்க, ஸோ கோல்டு ஃபிஷ் கீரை வரும். இப்போ நீ ஏஞ்சல் ஃபிஷ் வைக்கிறீங்க, கீரை வராது. அப்ப ரெண்டு ஃபிஷ் தானே, ஏன் கீரை? அப்ப உங்க சிஸ்டம் தப்புன்னு சொல்லக்கூடாது. இப்போ நீ எந்த ஸ்பீசிஸ் வந்து அது கூட காம்பினேஷன், ஏன் அதுக்கு காம்பினேஷன் சொல்றேன்னா, கோல்டு ஃபிஷ் will release more waste, metabolic waste. So that waste may be a cover for spinach, கீரைக்கு. அப்போ இதுல வந்து நான் சொன்ன ஏஞ்சல் ஃபிஷ்ல வந்து மெட்டபாலிக் வேஸ்ட் கம்மியா வரும். அப்ப கீரை வரலையேன்னு சொல்லக்கூடாது. அக்ரிகல்ச்சர் சிஸ்டம் எல்லா சிஸ்டத்திலையும் அந்த எங்க அந்த லேக்குலா இருக்கு, எங்க இஷ்யூஸ் இருக்குங்கிறத நம்ம ட்ராக் பண்ணி அதை சரி பண்ணணும். அப்படி சார் சொன்ன மாதிரி எஸ்ஓபி போறது கொஞ்சம் கஷ்டம். ஏன் கஷ்டம் அப்படின்னா ஒவ்வொரு ஏரியாவுக்கு வெவ்வேறு மாதிரி செட்டப் தேவை, வெவ்வேறு மாதிரி பேராமீட்டர்ஸ் இருக்கு. அப்போ என்ன இது போடலாம், இந்த ஆவரேஜ் ஆப்டிமம் இது இருக்கணும்னு போடலாம். சரி, அதனால இது சரி பண்ணணும். பட் அந்த அளவுக்கு நம்ம என்ன டிமாண்ட் போகல வரும் ஃபியூச்சர்ல. பெஸ்டிசைட் வேண்டியது இல்லை. இப்போ பாத்தீங்கன்னா எங்களுக்கு எந்த ஒரு இஷ்யூஸ் வரலையே, எல்லாம் க்ளோஸ் க்ளோஸ்.
சார் ஓவர் பீரியட் ஆஃப் டைம் இப்போ நம்ம அதை கமர்ஷியலைஸ் பண்ண போறோம், லார்ஜ் குவாண்டிட்டில பண்ண போறோம்னா எங்க இருந்தா
நம்ம ஓபன் என்விரான்மென்ட்ல பண்ணா ஓகே, க்ளோஸ் என்விரான்மென்ட்ல பண்றோம். இப்போ சப்போஸ்
கிரீன் ஹவுஸ்லயே நம்ம பிளான்ட் வளர்த்தாலும் ரெண்டாவது தடவை நீங்க கல்டிவேட் பண்ணும்போது எங்க இருந்தாலும் பூச்சியோ மைக்ரோபோ வரதான் செய்யும். அதே மாதிரி தானே இருக்கும். இட் இஸ் a natural law.
அதாவது என்னன்னா அதாவது இது கண்ட்ரோல் என்விரான்மென்ட்ல நாம என்ன பண்ண முடியும், ஈஸியா மானிட்டர் பண்ண முடியும். இது ஒண்ணு. ரெண்டாவது இப்போ ஃபார் எக்ஸாம்பிள் ஃபில்ட்ரேஷன் சிஸ்டம்ஸ் ஸ்ட்ரென்தன் பண்ணலாம். ஃபில்ட்ரேஷன் சிஸ்டத்தை ஸ்ட்ரென்தன் பண்ணாவே மெஜாரிட்டி ஆஃப் த டிசீஸ் வந்து உங்களுக்கு ரெடியூஸ் ஆகும். இது ஃபிஷ்க்கு மட்டும் இல்ல, பிளான்ட்க்கு சரி. ஆமா, பிளான்ட்க்கு தான். சோ அந்த மாதிரி அதாவது அக்ராஸ் அக்ரிகல்ச்சர் சிஸ்டத்தை விட இந்த சிஸ்டத்துல இன்செக்டோ மற்ற டிசீஸ்னுடைய இஷ்யூஸோ ரொம்ப கம்மி. அப்ப என்ன சிஸ்டம் வந்து பெஸ்ட்னு தான் பார்க்கணும் மொழிய. 1 or 2 டிசீஸ் வரதால இந்த சிஸ்டம் ஃபெயிலியர் சொல்ல முடியும்.
சிஸ்டம் ஃபெயிலியர்னு நான் கேட்கல. நான் கேக்குறது இப்போ எக்ஸ்பெரிமெண்டலா ஒரு தடவை வளர்க்குறோம், ஓகே, ரெண்டாவது தடவை வளர்க்குறோம், அதுல வரல, மூணாவது தடவை நீங்க அதுல இருந்து
சடன் வந்து அதுக்கு நீ கேக்குறது எங்க வரீங்கன்னு புரியுது. என்ன எங்க வரீங்கன்னா, சார் நீங்க ஆர்கானிக்னு சொன்னீங்க, எப்பயாவது பெஸ்டிசைட் ஒரு வேர்ட் நான் யூஸ் பண்ணணும்னு பாக்குறீங்க, பண்ண மாட்டேன். அப்ப நீங்க என்ன பண்ணணும்னா சிஸ்டத்தை ஷட் டவுன் பண்ணணும். ஒரு எனக்கு வந்து இந்த மாதிரி கண்ட்ரோல் சிஸ்டம் வந்து ஒரே சிஸ்டம் வச்சு மெயின்டைன் பண்ணக்கூடாது. ஆல்டர்னேட்டிவ் சிஸ்டம் வச்சுக்கிட்டே இருக்கணும். 2-3 சிஸ்டம் எங்க இஷ்யூஸ் வந்து அவங்க டக்குனு ரீப்ளேஸ் பண்ணலாம். ஃபார் எக்ஸாம்பிள் வந்து இந்த ரூம்ல இப்ப குவாரண்டைன் மெஷர்ஸ் இருக்குல்ல,
ஆனா நம்ம யூஸ் பண்ற செட்டப் காஸ்ட்லியரான
குவாரண்டைன் மெஷர்ஸ் இல்லையே. இப்ப அந்த சாமந்தி பூரா என்ன காஸ்ட்லி, ஒரு டியூப் தானே. அப்ப எங்க இதுல நாங்க வச்சிருக்கவே அந்த அக்வாபோனிக்ஸ் அதெல்லாம் பெரிய காஸ்ட்லி ஒன்று கிடையாது. எப்ப அந்த திங்க் வந்து நம்ம காஸ்ட்லியா திங்க் பண்ண காஸ்ட்லி இல்ல, நம்ம கிட்ட இருக்கிற ஓல்டு மெட்டீரியல் யூஸ் பண்ணி நம்ம அதை மாத்திக்கலாம். எக்ஸிஸ்டிங் மெட்டீரியல் நீ கன்வின்ஸ் ஆகல, கேள பண்ண ஒண்ணும் இல்ல.
நம்ம இப்போ சின்ன செட்டப் மொட்டை மாடியில வைக்கிறோம்னா வேஸ்ட் மெட்டீரியல்ஸ் பண்றோம், ஓகே சார், கமர்ஷியலைஸ் பண்ண போறோம், ஒரு கம்பெனி தான் அதை பண்ண போகுது அப்படின்னா they'll be using it in hectares. So how will they shut the system down such that there is
if I can say whatever system actually whatever system you are using, even we can say nearly 10 years guarantee for the PVC pipe. சரிதானே? அப்போ 1 crop முடிச்சுட்டு நான் இவ்வளவு இன்வெஸ்ட் பண்ணேன்னு சொன்னா லாஸ் தான். அது வந்து 10 இயர்ஸ் இருக்குல்ல. மல்டிபிள் கிராப் வேற ஒரு சப்ஸ்ட்ரேட் ஒன்ஸ் இன்வெஸ்ட்மெண்ட் காஸ்ட் வந்து நம்ம காஸ்டா போடக்கூடாது. இன்னைக்கு எக்கனாமிக் ஒர்க் அவுட் பண்ணும்போது அது ஒன் டைம் இன்வெஸ்ட்மெண்ட், புரியுதா? அதுக்கப்புறம் எத்தனை நம்ம மல்டிபிள் கிராப் கொண்டு வர போறோம், என்ன வெரைட்டி ஆஃப் கிராப் கொண்டு போறோம். நான் அப்படியே சொன்னேன், ஃபார் எக்ஸாம்பிள் முன்னாடியே சொன்னேன், இப்ப இவ்வளவு பெரிய சிஸ்டம் நீ டெவலப் பண்ணிட்டு அதுல வெண்டிக்காய் கத்திரிக்காய் வச்சு ரெஸ்ட் ஸ்ட்ராபெரியோ வேற ஏதாவது ஃப்ரூட்ஸ் வச்சுன்னா அதுக்கான ரேட் கிடைக்கும். அப்போ how we are going to invest according to that which plant species we are going to select, thinking is one. And இந்த மாதிரி அந்த பிளான் பண்ணிட்டா there is no loss. And another thing is a one time investment. இப்போ சாட்டிஸ்ஃபைடா? அப்போ இன்னொரு விஷயம் என்னன்னா, இப்போ சப்போஸ் குவாரண்டைன் பண்ணணும், ஷட் டவுன் பண்ணணும். ரெண்டு தான் நேச்சுரல் வே, பெஸ்டிசைட் போகக்கூடாது, ஆல்டர்னேட்டிவ்ஸ் தான் இருக்கும். அதுக்கு குவாரண்டைன் பண்ணும்போது நீங்க ப்ராப்பரா அது என்னது ஃபியூமிகேஷன் அது சானிடேஷன் ஒர்க் என்னது 100% என்ன பண்ணுவீங்க, குளோரினோ இல்ல வேற ஏதோ போட்டு ப்ராப்பரா அதை டிஸ்இன்ஃபெக்ட் பண்ணிட்டு ஓப்பன் சன்லைட்ல வச்சு ஒரு 2 or 1 month கேப் கொடுத்து திரும்ப நீங்க போனீங்கன்னா டிசீஸ் ரெடியூஸ் பண்ணுங்க. அக்வாகல்ச்சர் இண்டஸ்ட்ரியில ஆரம்பிச்சதுல இருந்து 90s 80s இருந்து டிசீஸ் தான். இண்டஸ்ட்ரி மூணாங்க, ப்ரொடக்ஷன் இன்க்ரீஸ் ஆகுது இல்லையா? Students infection.
Yeah, thank you, students. Please, we have question during the lunch session. You may ask questions. Okay. The next person is waiting. Thank you so much, Dr. Raja sir, for your wonderful presentation. Thank you so much.
We have one more plenary lecture by Dr. John Paul. Dr. John Paul is an assistant professor in the Department of Zoology, St. Joseph's College, Bangalore, and teaches classical zoology, allied zoology, embryology, conservation biology, immunology, and animal ecology. He completed his BSc, MSc, and MPhil zoology in Loyola College, Chennai, and earned his PhD from the University of Madras. His thesis entitled Beta-1,3-glucose binding protein mediated cellular immune responses in the marine mussel Perna viridis. His area of specialization includes protein chemistry, molecular immunology, and immunobiochemistry. He has worked as a project fellow in Noni Foundation Collaboration Project, Junior Research Fellow in UGC Major Research Project, and UGC DSR program at the University of Madras. He has an ongoing research project development of magnetic particle enzyme immunoassay for diagnosis of leptospirosis, sanctioned for 5 lakhs. He has served as a PhD Viva Voce external examiner, delivered invited lectures, attended and presented papers in 27 conferences, published research articles, and submitted 4 sequences in GenBank. He has also organized 12 conferences and workshops and is skilled in biodiversity related groupings, identification markings, and labeling of organisms related to gastropods. He is an active member of Zoology Association, Loyola College, Entomology Academy of India, Natural Sciences Association, St. Joseph College. We are indeed honored to have you with us, sir. May I now request Dr. John Paul to deliver his plenary lecture.
Good afternoon, everyone. People are tired. Okay, lunchtime also. Right, I'm given only like half an hour to present my work. I'll try to finish by then. Okay, but the work which is being put forth over here for the presentation is a bit, what to say, unlikely. Okay, you may not understand, I don't know, right? Whatever it is, if you have doubts, clarifications with the presentation, please do feel free to ask me then and there. And it's my great pleasure to come here to St. Joseph's College, Palayamkottai. And this is my 4th visit. I've already come as I don't know, participant. Actually, cannot call it as a student for a refresher course. So before 4 years, and after 4 years, I'm here now. Okay, to present before you. And this is my actually my PhD work entitled Beta-1,3-glucose binding protein mediated cellular immune response in the marine mussel Perna viridis. And before I could move on, I want to put forth one important thing for the people who are intending to do research. This work has a background of 30 years. Okay, 30 years. And I'm the 3rd one to get a degree in the same field. And before that, my guide, he had been working from the 1970s. Okay, this is the thing I wanted to put forth. And therefore, even with a small lineage, you can carry out a very good research work with a good publication, and which is supposed to be useful to the human society. Okay.
Now, I don't know if you people know are aware of the immune functions or the immune system of the human beings as well as the lower grade animals. If you need to talk about the immune system as such on the whole, that will have two components, cellular component and the humoral component, and that's what is being focused over here. And this is not applicable only for the molluscans, even for the human beings is the same. Even for the with a bit of what to say development or differences between the molluscans and the human beings. That's what is shown over here in this particular slide. Next one. Okay, and as an introduction, let me tell you orally. There is one particular protein called as beta-1,3 glucan binding protein, which has been isolated from the plasma. You know the plasma serum. Okay, in the invertebrates, we have something called as the serum, which contains no blood cells. Okay, we have two different components, the humoral component and the cellular component. And the humoral component has this particular protein called as beta-1,3 glucan binding protein that was isolated and then purified, and we went on to the characterization process. On one side, the work overall outline, let me tell you, and then I'll continue. Then this was on one side, and the next part, what we had done is the cellular component we took into account. My main part of the work is with the cellular component. We call the cellular components which is being present over there, the invertebrates as hemocytes. Now in the muscles, in the molluscans, you have three different types of hemocytes called as granular, okay, and hyaline cells. Okay, hyaline cells, semigranular cells, and granular cells. These three different categories of the cells have been found over there in these molluscans. This is on one side. Now you have the protein, you go for the production of the polyclonal antibodies against this particular protein using the mice. I have all the results. Due to the time constraint, I just want to give an abstract of the work and then continue with the result alone. Right? Now you have on one side the protein purification, and on to the particular protein you have the polyclonal antibody production. Okay, on one side. Now after producing the antibody, you need to go in for the purification of IgG alone. So you have in the polyclonal antisera, you have all the four different components. You have IgG, you have IgM, you have IgD, you have IgA, you have IgE also. You have all the components of the immunoglobulins. Okay, we go only for the purification of IgG, which is supposed to be the most conduct part, at least for about like 70-75% of the immunoglobulin. So you go for the purification of this IgG. You couple that with the immune stimulants, we call that as, okay, those results are onto the third part. This is on another side. Now, these coupled IgG antibodies, right, are to be tested with these hemocytes about the phagocytosis process and the encapsulation process. We hypothesize in this particular work that there is something which actually triggers the activation of these hemocytes to do the phagocytosis process. We call something as opsonin, and this particular protein functions as an opsonin. Okay, I hope you understood the ground reality of the work.
Can you repeat the last?
See, there is something called as opsonin. Okay, we hypothesize that this particular protein, beta-1,3 glucan binding protein, has the opsonic capacity of activating or promoting the phagocytic effect of the chemocytes. See, we have the macrophages, WBCs, in our human body. That macrophages, WBCs, they do the functioning of killing the invading pathogens. It could be anything. The same mechanism is employed over here in these molluscs. They have hemocytes. They also naturally kill the invading pathogens But add this particular protein, adding this particular beta-1,3 glucan binding protein, the effect of this phagocytic condition is going to be more, one step ahead. Is that clear? Okay, can I move ahead? Right. Now, one more point. Here, this particular work was started in the year 1992. Okay, and there were 3 papers being published with the same animal. And after that, that was continued from 2000 to 2007. And I took over from 2008. Okay, so 3 theses, 1 more is pending. Okay, 1 more loophole is there for this particular work. Okay, if any one of you are, if any one of you are interested to do this particular work, you can contact me. Okay, I'll give you the full details. You can work on it. Fine. Okay, so you can skip the slides. Perfect. Okay, and therefore the objectives of this present study, okay, is to elucidate the functional heterogeneity of the different hemocyte morphotypes. I told you about 3 different hemocyte morphotypes. The first point is that. The second one is to localize the BGBB, the beta-1,3 glucan binding protein in the humoral as well as the cellular compartments. Okay, in the hemocytes itself. Then to find out or to demonstrate this particular protein, whether it is being present over there or not in the humoral factor as well as in the cellular factor. Okay, right here we have another hypothesis that we still even till today we think it like the cellular compartments of these hemocytes, they also contain certain molecules which are relevant to this beta-1,3 glucan binding protein. Okay, even now we have hypothesized, even after doing so much of a work. Okay, for this, one more PhD thesis is required to come up with the result. Okay, second, perfect. Okay, this is the animal model. Okay, this is also known as the green muscle. Okay, the marine one, right? Mostly adhering to a substratum. Okay, mostly it's found in the boats. Okay, rocks. Okay, in the shore areas of the marine environment. Yeah, next one. Right. The first part is supposed to be the previous slide, sir. The isolation and purification of beta-1,3 glucan binding protein. This is supposed to go way back to almost like 10 years. Okay, from now 10 years back, right? And therefore I'm not going in detail about it. Just next slide, sir. Now the preliminary studies which have been conducted, whether to identify this particular protein is in function or not. For that, we did an agglutination survey, right? You know agglutination. Okay, right. The second one, and this is the purification process on the board. Okay, which is put forth over there along with the. Right, okay, native as well as SDH phage analysis. Right, the next one. Okay, then after purification, we also went for the agglutination titer. Okay, assay. Okay, and these were the results. And the result of these purified beta-1,3 glucan binding protein was an outstanding one. Okay, outstanding one when compared to the whole plasma. See, whole plasma is supposed to contain most of the protein content. Multiple proteins or multi-proteinase activity can be found over there in the whole plasma, like our whole blood. Whole blood will contain all possible sorts of proteins. Okay, if you need to take out only one particular protein in the blood, you need to go in for the purification. And then after purification, you need to go in for the checking out of whether it really is that particular protein or not. Okay, and therefore this study was done. Okay, then we moved on with the generation of the polyclonal antibodies and purification of the anti-BGBP, IgG molecules, and then we went it for the FITC anti-BGBP conjugates. Okay, to do our the furthermost study, then to the later stage. Yeah, next one, sir. Okay, and this is a procedure normally being followed for the production of the polyclonal antibody. The next one. Okay, and after producing the polyclonal antibody, you need to confirm it, right? Whether if there is an antibody being produced,
and if that antibody being produced, which is supposed to be corresponding to this particular protein or not. And therefore, you have the double immunodiffusion method, you have the ELISA method also, just for the confirmational test. Right, next slide. Okay, and then we moved on with the purification of IgG, as I've already told you. And this is what the purification peak, okay, in the protein A, CL agarose column we used. Okay, and this was the peak we got. And then we need to confirm that again with something called as the Western blot analysis. Okay, if it is corresponding to that particular protein or not. Okay, and after that only it was confirmed that this particular protein has an effect over, and it was induced into the mice. Okay, the antibodies were produced, and the IgG molecules which were produced in the antibodies were corresponding to this particular protein. Yeah, next slide, sir. Okay, and the second part, okay, we moved on to a study of what the cellular immune responses of the beta 130 glucan binding protein, okay, from the marine muscle pernabratus. The second part. Next slide, sir. Okay, and before going into study about the cellular components, you need to make something called as a hemocyte, okay, a monolayer. Hemocyte monolayer we call that as, and this is a formula for the hemocyte monolayer to be formed. Okay, just to study about the different cell types along with the viability as well as the phagocytosis assays. The next one, sir. Okay, and these were the photographic images of the phase contrast microscope and the DIC. Okay, differential intentional configuration. Okay, these were the photographs which were taken during our study. Okay, just to find out whether there are hemocytes available and to what extent they are available. Okay, to what category they are available. We just wanted to study about it, and therefore we have isolated the cellular component alone. The next slide. Okay, and then this is the whole part. Okay, I've already told you, like, there are 3 different hemocytes being present over there in the whole hemolymph of this animal. Now, our next aim was which of the hemocyte has the binding capacity, which of the hemocyte will be having a kind of a phagocytic activity more. We wanted to find out that, and therefore we took the hemolymph. Okay, we wanted to separate all the hemocyte morphotypes, all the 3 different hemocyte morphotypes, and therefore we used something called as the discontinuous density gradient of Passcode. Okay, the Passcode gradient were used to separate the different hemocyte morphotypes. Okay, and this is the formula which is used for the separation technique using the Passcode gradient. Next one, sir. Okay, and this is how it is going to look like. I don't think so it is visible. Okay, can you see the layers? 1, 2, 3, 4. Can you see the layers? Okay, it should be visible, right? Now, in this particular interval of these layers only, you can identify the different types of hemocytes. Okay, all the 3 basic ones, the hyaline, semigranular, and granular hemocytes. Okay, with the density of the Passcode gradient, they would also be coming down. Now, before going into it, let me give you another clue about these hemocytes. Okay, the hemocytes are identified based upon one particular character, based on the presence or absence of granules. Granules on the surface of the hemocytes. If there are the hemocytes filled with completely granules, then they are going to be known as granular hemocytes. If it is partially filled, it's going to be known as semigranular. If there is few or none granules, it's going to be known as, okay, hyaline hemocytes. Okay. Now, based on the number of granules being present over there in the hemocytes, right, the density or the weight of the hemocytes is going to be found. That's what is shown here. Okay, now whatever is going to be found over there in the layer 4 are all the granular hemocytes, the third one are all semigranular hemocytes, and the second one are all the hyaline hemocytes. Okay, right. Now we have identified next one. Okay, and then we wanted to find out the total and differential hemocyte count also. Okay, total and differential hemocyte count on the whole before the separation technique to be used, and after the separation technique being used. Now, when you take the whole hemolymph, okay, you have the unfractionated hemocytes as 3.29 and then isolated as 2.6, which is going to come down. Which drastically comes down, actually speaking, when you take for 100%. Okay, now again, you see, after the hemocytes being separated using the peptide gradient, okay, you see the total cells. Okay, everything is supposed to constitute for 100%. Everything is supposed to concentrate for 100%. The hyaline cells, semigranular, and granular cells. Right. So next one. Okay, and after the separation, after the separation technique was used using the peptide gradient, this is how the cells look like. This is how the cells look like. Okay, I've already given you the pre-information. Right. The next one. Right. Now here we want to study about the viability test also. Okay, viability. So when you need to study about the phagocytosis, which is going to take for about like at least half an hour to 45 minutes to 1.5 hours, you need to study about viability of these hemocytes. When you place them onto a glass slide, okay, they should be alive. Only if they are alive, they can do the engulfment or the phagocytic activity. Otherwise, there's going to be nothing. And that's where the outstanding work of this viability was studied. Okay, the next one. Okay, and then we also wanted to study about the flow cytometry content in order to find out what hemocyte to what extent is being present over there. And this is the result is that we got. Okay, more of the granular cells, very less quantity of the hyaline cells, and the medium of the semigranular cells. Okay, can you just go to the previous slide? Previous one. Okay, the previous one. The total number. No. You can compare this slide with the present one, what was shown over there. Yeah. Okay, you can compare that if you have any doubts. Right? Now, anyways, I'll leave the material here to the Department of Zoology. If you want further reference, you can always take and utilize it. Right? Right? Okay, this is the flow cytometric assay which was being done. Along with this, we have also done many other flow cytometric assays also. Next one. Okay, this is with the propylidomide just to find out the viability. The next one. Okay, and then this is with the FATC. Okay, FATC only with FATC. What is FATC? What's the abbreviation for FATC? Fluorescein isocyanate. Okay, right? So it's a coloring agent. That's all. Right? Now, only with FATC the hemocytes are being induced. How are they going to look like? Okay, and the next one, sir. Then FATC with BGBP, the beta 1,3 glucan binding protein, along with the FATC when it is being induced with the hemocytes. Next one, sir. Okay, now the FATC along with the other components are also being given over there for the study. The next one, sir. Come. Okay, then this is actually speaking about the phagocytic component of the hemocytes. The phagocytosis assay, the actual phagocytosis assay which was done over here. Right? This is for the unfractionated hemocytes. Okay, we have it for about like 8 different tests. Okay, inclusive of the control. Okay, in which the beta glucan binding protein mediated yeast, which was consumed to the maximum, which was utilized to the maximum for phagocytic activity. Okay, for about like 74% to 76%. The next one. Then, after getting this confirmed, we wanted to find out based on what category the beta-1,3 glucan binding protein is effectively activating or effectively functioning as an absorbent. Okay, and therefore we moved on with the dose dependent, okay, increased activity of the phagocytosis. Okay, and then we got to a conclusion, like, no, to a certain extent, the amount of VGP which was tested, okay, up to 75 microgram, okay, the activity was there. Apart from that, when you increase the quantity of the beta-1,3 glucan binding protein, the activity was lost actually. Okay, now we have all as saturation limit. If you get beyond a saturation limit, gone. Get the point? Okay, so we are all of us. Now, if you need to eat, you have to eat only to the maximum extent of how much your stomach can afford to. That's all. You cannot overload yourself. That's what has taken place over here also, and therefore the dose dependent study was done with the phagocytic activity. Next one. Okay, then we separated the hemocytes. After the separation of the hemocytes, we wanted to study with the individual hemocyte morphotypes, hyaline, semigranular, and granular. Okay, and with no effective cost of these hyaline cells, which have no granules being present over there. Okay, they also showed to a surprise, okay, that they do possess phagocytic effect. Okay, right? They do possess phagocytic effect. This was to a surprise for us. Okay, the next one. Okay, now the semigranular cells and granular cells. Next one. Okay, they all show. Okay, they all show the same set of an experiment which I've already told you. But hyaline cells were actually for a surprise. Okay, they also had an activity. Right, the next one. And this is the overall table which was given over there for the explanation purpose of the previous slides. Yes, next one. Okay, then after the separation of these hemocytes, we also wanted to check out for the dose dependent activity on the three different types of hemocyte morphotypes, hyaline, semigranular, granular, and this is what is being shown over here in this slide. And you see again here at 75 microgram. Okay, at 75 microgram of the beta glucan binding protein, what happens? Okay, both with with all the three different types of hemocyte morphotypes. The next one. Okay, then this is the actual diagram which shows about the phagocytic activity of the different hemocyte morphotypes. Okay, when coupled with yeast, trypsinized yeast. Okay, do it like you have the protein, you have the yeast, you have the trypsinized yeast, right? When you coat that with the protein, okay. Now, if you consider this as the yeast cell, the coating, the protein coating of this particular protein will be like, okay, enhancing the size of the yeast or the foreign substance, the foreign material, and therefore that makes a way for the hemocytes to identify, to recognize that there are some invading pathogens which have got entered, and therefore we need to go fight and kill. For that purpose, it's being used, and therefore we still speculate that okay, there are layouts for this particular protein to be present over there as receptors, or they might have receptors on the surface of the cells. That's where the activities will taking place. The next one. Okay, and then we move on with the third and important component of our study, right? Identification of BGP receptors and binding proteins and various isolated or fractionated hemocyte morphotypes using immunocytochemical and fluorescent dye. Okay, this was our last work which was done. Okay, to complete my PhD work. Okay, the next one. Okay, and this is the first one known as the immunocytochemistry. Okay, immunocytochemistry. You would have done at least one or two of you would have done immunohistochemistry. Okay, and this is immunocytochemistry since it's a study of cell. Okay, you just need only the dye to study, right? And this is the answer is that we got. Okay, with the cytochemistry that only the granular cells were so much effective to this particular receptor. Okay, the next one. Then we moved on to study of study with respect to FITs. Okay, the fluorescein isothiocyanate. Okay, here also we could come to a conclusion that compared to hyaline and semigranular cells, the granular cells had so much of an effect. Okay, and therefore we came to a conclusion that in the cell surface of each of the hemocyte morphotypes, you have the binding sites for this particular protein called as beta-1,3 glucan binding protein. Okay, and these are actually speaking the preliminary work which has been shown, which has been given. There is again a lineage wherein we need to identify if at all in the surface the receptor being present, how it is, where it is, what it is, and we need to go into the gene sequencing, protein sequencing, okay, DNA sequencing, to get into the molecular level to come into a conclusion or for a confirmational test. And this is going to go in for another PhD. Thank you so much. Any questions? You better go for lunch. Okay, you ask me there if you have doubts, clarifications. It's already time. Thank you.
On behalf of the organizing committee, I thank you once again, sir, for knowing the time and finishing it on time, and for sharing your detailed explanation on your PhD research work on beta-glucan binding protein. So now we will go for lunch. The lunch is arranged in the main block, where in the Department of Zoology. The Department of Zoology is in the second floor. And I request our department students to guide the outside participants to the lunch hall. Soon after the lunch, those who have oral and poster presentation will remain in the Department of Zoology itself. We have arranged smart classroom there for oral presentation and for poster presentation. The other participants who are not involved in either oral or poster presentation can come back to the same hall at 2:15 p.m. We will have one more lecture here after the lunch. Thank you so much. An announcement, please. Since the prayer is going on, we request you to remain inside the hall for 5 more minutes. Soon after the prayer ends, you can leave. Because we are not supposed to move inside the college when the prayer is on.
Dear participants, the session will start at 2:15 p.m. They kindly wait. Now others will join, then we'll start. Marimut sir.
Madam, the label வந்து
Hello sir. Hello sir. வணக்கம்.
Okay.
Sir, கேக்குதா? Dr. Alavaraj?
Hi sir, are you audible?
Audible sir. Can you hear me sir?
Yes sir, yes sir. Sir, kindly share your PPT on the slide, sir. Just to check it.
Okay. Okay, sir. Can you see? Can you see the slide?
Yes, sir. It's visible, sir. Yes.
Okay. How about the audio, sir?
Audio is audible, sir.
Okay, fine.
Clearly audible, sir. Okay.
Okay.
Sir, wait for a couple of minutes. I will start at 2:15, sir.
Dear participants, shall we start the meeting? Sir Marimuthu sir.
Yes, sir. Please proceed. Yes, sir.
I am happy to introduce Dr. Marimuthu, Professor of Department of Biotechnology, Faculty of Applied Sciences, Amist University, Bidang, Geda, Darul, Aman, Malaysia. He teaches biology of invertebrates and vertebrates, aquaculture, biostatistics, research methodology, environmental biotechnology, animal biotechnology, human physiology, genetics, business management. He completed his BSc Zoology, specialization in fisheries, MS University, MSc in environmental biotechnology, specialization in aquaculture biotechnology. MSc Zoology in Madurai Commerce University and PhD in Zoology Environmental Biotechnology. He earned MBA from MS University and Diploma in Industrial Pollution and Control from Annamalai University. He worked as a junior research fellow and project instructor in Center for Aquaculture Research and Extension, St. Xavier's College, senior research fellow in ICAR Bhubaneswar, and postdoctoral fellow in the School of Biological Sciences. His research specialization includes breeding and seed production of commercial important air-breathing and threatened fish species, larval rearing of air-breathing fishes, nutritional requirements of freshwater fishes, fish immunology and immunomodulators in aquaculture, breeding and culture of leech, and toxic effect of pollutants in fishes. He has had the privilege of visiting China, Singapore, Taiwan, Malaysia, Indonesia, Sri Lanka, and Thailand. He is an active member of many professional bodies and university committees like National Academy of Biological Sciences and Asian Fisheries Society, appointed as an external examiner for many PhD Viva examination, recipient of Excellent Researcher Award in Biotechnology, editorial member of Indian Journal of Natural Products and Resources, International Journal of Histology and Fisheries, Journal of Arid Land Agriculture, supervised more than 70 UG and PG projects and 3 PhDs. He has received grants for 6 projects worth of 8,20,000 in Malaysian ringgit, equivalent to rupees 150 lakhs. He has for his credit 120 research publications in national and international journals, 10 articles in popular magazines and bulletin, 2 books and proceedings, 5 chapters, 40 conference and seminar presentations, and 6 submissions in Gene Bank with accession numbers, delivered 42 invited talks and lectures. His works are cited more than 2,000 times with an H index of 29 and I-10 index of 59. In the past 6 years, he is known for conducting around 30 workshops exclusively in statistics. We are happy to have him as a resource person of this section. Over to you, sir. Now you can present.
Yes, sir. Thank you. Thank you, Dr. Alagraj. Dr. Alagraj, right?
Yes, sir.
Thank you so much, sir, for the nice introduction, and very good evening. I think India is a good afternoon. Good afternoon, participant. And first of all, thank you so much for giving this invitation, for joining this interesting topic of the forum, recent trends in aquaculture. So I'd like to congratulate all the organizing committee members. They have chosen a very interesting topic for the discussion and today's discussion. So the topic I have chosen for today's presentation is about breeding and seed production of naked fishes, Channa striatus and Channa punctatus, very common freshwater fish species in India. And another fish is African catfish, exotic species, very common in Malaysia. So I have been working on this fish for many years, funded by DST India and during my PhD time. And in Malaysia also, I have been working on the African catfish. So I have put together as a simple presentation. So the technology, whatever we have gained, we have developed. So I'm going to share with you the simple breeding method and along with my research publications in this presentation. So the background you see here, this is the university where I'm working for the last 17 years. I started as a lecturer and now as a professor. So working in the Department of Biotechnology. So the university actually is from 430 kilometers away from Kuala Lumpur Airport and around 60 kilometers from Penang, the closest international airport. And basically we have a medical university, it's a medical university. We have medical pharmacy, dentistry, physiotherapy, nursing. And then we have a BSc, MSc, PhD in biotechnology. And recently we have got a BSc in bioinformatics as well. And then, so already Dr. Alagraj has been introduced me and introduced about me. I'll be covering about my research area and what is air-breathing fishes and what are the fish species we have in India. And then some of the characteristic features of the snakehead fish. In Tamil, we used to call viral mean, their diversity, what do they feed, and a little bit about their reproductive biology, and what are the constraints we are facing in India for culture of these snakehead fishes. And then let me also touch the mechanism of breeding of fishes, generally, the general physiology of the breeding mechanism, the hormonal mechanism, the reproduction physiology, little bit I'll cover. Then I will cover about the breeding and reproduction method of snakehead fishes, spotted snakehead Channa punctatus, and the striped snakehead Channa striatus. And then the breeding methods we have developed for African catfish in at my university. And what shall I do? So when you look at the air-breathing fishes, in addition to gills, the air-breathing fish species do have the accessory respiratory organ. And there is a diversity of respiratory organ in different air-breathing fish species. Lungs, gas bladder, buccal cavity, bronchial, opercular chambers, esophagus, stomach, intestine, gills, and skin serve as air-breathing organs. So these fish species has been increased dramatically. The culture of the air-breathing fish species globally has been dramatically increased. So reason, they are high-value fishes. And then you also can sell the fish in a live conditions. Without water also they will survive because of the nature of air breathing. So they can breathe atmospheric air for their respiration in addition to the gills. So without water also they will survive. So when you look at the global scenario, 8% of the global aquaculture production from air breathing fish species. And I will give you some of the examples of air breathing fishes. And some may be familiar to you, the participants. And like a marble goby, striped catfish, Pangasius species, and then snakehead fish, eel we have, snakehead fishes, walking catfish, Clarius patragus, African catfish, all right, and some of the even singi, Heteropneustes fossilis, very common, and also highly edible fishes, marble goby, etc. So they do have different types of air breathing organ. For example, snakehead fishes, they have a suprabronchial chamber, whereas an African catfish also similar to that. So they have kind of what you call inside there. In addition to the lungs, they do have kind of brush-like organs, and richly supplied with the blood vessels, and utilize the atmospheric oxygen and exchange the carbon dioxide. Very interesting to work with this kind of air breathing fishes. Easy to work with. Not like somewhat very hardy because of the nature. So these are some of the air breathing fish species. This is a marl. In Tamil, we used to call viral mean. Scientifically, we call them snakehead fish, Channa striatus. This one actually is taily mean in Tamil. In English, we used to call them singi. Scientifically, we call them Heteropneustes fossilis. This is Clarius caripinus, sorry, Clarius patragus, and this is a spotted snakehead marl. So this is also one among the species of Chinidae family. And this can grow even up to even 1.5 kg, 2 kg. So this can go up to even 300 grams, very minimum size. There are 30 over snakehead species available in globally. And a couple of species also available, very common in Tamil Nadu and southern parts of India. And this is a snakehead fish. Very highly delicious fish and healthy fish also. And it has got a lot of medicinal value as well. Party fish in some parts of Malaysia, they utilize as a medicinal value, the particular fish. Reason, they do have kind of some amino acids and fatty acids. It can foster the wound healing process, stimulate the wound healing process very fast. So people, they go for surgery. When they come back, doctors also will advise, please consume this particular fish species so that your wound will be healed very fast. So I have already mentioned there are 30 species of naked fish in Asian countries. They belong to the family of Chanidae. They are very delicious fish species, and also highly demanded food fish. And also they have a good market value globally. In India, sometimes you may reach 600 rupees. And hence there is a good consumer preference also. And I will show you some of the important naked fish species. Channa striatus, available in Malaysia, China, Thailand. And this is a punctatus spotted naked. China also available, India also available, very common. Especially they prefer shallow water. They can also withstand even low dissolved oxygen level. And some are edible fish species, some are what you call colorful ornamental value. Even Channa Kachua, used as what you call ornamental fish species, available in Indian waters. And these are some other snakehead fish species. Many more, 30 over species. So when you look at the characteristics of snakehead fish, Ikan Harwan, we used to call in Malay. In Tamil, we can call them as a viral mean. Scientifically or commonly, we can call as a striped snakehead. And they are native fish species of African country and also Asian countries. One among the highly demanded freshwater fish species. The prices from 400 to 600 rupees. And the tissue is well known for taste. And they have a very less intramuscular spine, high nutritive value, and also have medicinal values, and recommended as a diet during people, they go for any kind of recuperation, convalescence. Reasons, it contains amino acids like glycine, arachidonic acid, polyunsaturated fatty acids. This will promote the wound healing process and promote the prostaglandin synthesis and promote the wound healing process faster. The fish species are very hardy, breathe atmospheric oxygen for their respiration. They can tolerate even low dissolved oxygen level. This is more advantageous for culturing this kind of fishes. They can survive in without water also for several days until they do have a body moisture. Because of this nature, you can also stock 75 to 460 fish species in 1 meters per area. High stocking density also, this can be cultured. Basically, they are carnivorous, and they also do have one drawback, they are cannibalistic. So always healthier one will eat the weaker one. Bigger one will eat the smaller one. And that cannibalistic nature even started in before the 30 days after hatching. So then, because of the carnivorous nature, the fish also can be used as a biological control, controlling mosquitoes in the natural water bodies. So this has been used as a biological control organism. They eat mosquito species, mosquito larval forms. So they are suitable for sewage or where the oxygen level is low, any kind of oxidation pond. Other fish species, they may not survive. So this kind of fish species can be used for culture of even sewage water aquaculture. Look at the flesh, very delicious. Very delicious, only one straight bone, intramuscular bone, very less wastages, and high commercial value and very good taste also. And these are the 4 common species available in Tamil Nadu: Channa punctata, Channa marulius, Channa Kachua, and Channa striatus. Then why it has not been commercially cultivated globally and also in Asian countries? Bangladesh, Sri Lanka also, Malaysia, Thailand, the snakeheads are available. Why it is not much? Reasons: availability from the wild is very, very limited. We cannot rely only on the wild seeds. And availability of the broodstock also in the wild is limited. And hatchery technology not like other species like the carps. It's not available. No government hatcheries, no private hatcheries. They are producing naked fishes. And then farmers also, they do have a lack of the knowledge about the feeding and breeding. And additionally, they are also cannibalistic. So the maximum 1 kg fish can produce 20,000 to 40,000 progeny. So comparatively, maybe at the end you may get 50% survival rate. 50% survival rate. And do have a commercial market value. And these are the constraints. The naked fish species not very well popular, but nowadays is getting much prominence. Many farmers they have started. Even the naked fishes also they can consume the commercial pelleted feed. Nowadays some companies also they come up with the pelleted feeds, formulated pelleted feeds they are consuming. So when you look at the feeding biology, fundamentally they are carnivore. They're feeding mainly on animal materials, animal food right from the beginning, from the larval stage. They spawn and they only feed on almost exclusively zooplankton like copepods and cladocerans, the fry and fingerlings. Juveniles they consume aquatic insects. The fingerlings they consume aquatic insects and the small crustaceans, fishes, small fishes. Adult they consume small fishes, frogs, crustacean, insect larvae, etc. When you look at the reproductive biology, they are capable of breeding once a year. Some fish species, for example, in tilapia, every 1 month they are ready for spawning. Some fish species twice a year. So this annual spawner only once they do breed, that too in the monsoon season, June to September, or in October to December, during the rainy season. So female releases 1,500 to 20,000 eggs per span, one particular spawning, and it takes 24 hours for hatching after fertilization. And hormonal method also been standardized, inducing the breeding. So it takes 24 hours for hatching. And then it has been reported that after induced spawning also, the parent, they provide parental care. Meaning the parental fish always safeguard the young ones. They'll be very aggressive. They will protect the young ones for some time. Maybe it has been reported for 45 to 50 days. The parent always will be there. They go together in the natural water body. So these slides, I will show you the breeding methods have been standardized. There are different hormonal preparations have been used for breeding, especially naked fishes. The first one, by giving the conducive environment in the captive conditions. Put male and female together in an earthen pond with some vegetations. We need to give them quality feed, fish feed. And when they're mature, they reproduce during the spawning season, natural way. Another method, we can use the natural hormones like a pituitary extract and human chorionic gonadotropin. And we have some synthetic hormones like Ovatide, Ovaprim hormones are available. So when you go for where these hormones will elicit the functions, mechanisms. So when you keep the fish species in the natural conditions in the earthen pond, the mechanism will be like this. The hypothalamus receives the what you call the environmental stimuli. Hypothalamus releases GnRHA, a gonadotropin releasing hormone. So this hormone stimulates the pituitary to synthesize gonadotropin. Gonadotropin travels through the blood circulation, reaches the ovary in female and male testis, wherein stimulate the ovary to produce steroids. Steroids releases prostaglandins. Steroids is responsible for maturation of egg. Prostaglandins, it is responsible for ovulation. Ovulation means releasing of eggs from the ovary. Right? So this is a natural process. When you induce the synthetic hormones through the administration, it directly goes to the pituitary gland. Reason, the pituitary gland do have the receptors for the hormone, gonadotropin releasing hormone. So synthetic analog which are available in the market, they are gonadotropin releasing hormone, synthetic analog. It teaches and stimulates the pituitary gland, stimulate the gonadotropin. So when you inject natural hormones like a gonadotropin, human chorionic gonadotropin, and pituitary extract of fish, when you inject through the blood circulation, it goes to the ovary. So inducing synthetic hormones, it induces producing endogenous gonadotropin, whereas the natural hormone like a human chorionic gonadotropin, pituitary extract, they are called exogenous gonadotropin. Exogenous gonadotropin. It goes to the ovary, and the rest of the process is same. I hope some of the participants might have understood the mechanism. So this is the process normally, synthetic. So this particular slide, I'm showing you the induced spawning method. This is a broodstock. Normally 2:1, 2 male and 1 female. So we need to identify the healthy male and female fishes without any diseases. The female and male fishes must be matured. So 2:1 ratio, we need to identify. And then this way, the hormones can be injected, intramuscular injection. We have standardized it. Same concentration, 0.4 ml to 0.5 ml per liter, per kg of body weight, male and female. And after hormone administration, you need to slightly massage it on the particular injected site and release them into the earthen pond. You can also use HAPA. And then after 12 to 14 hours, you could see the spawning behavior. They start what you call reproducing. And 24 to 30 hours, the male and female, you could see the aggressive spawning behavior and releases egg and sperm. So fertilization takes place outside the body. You could see this is a fertilized eggs of snakehead fish observed under the microscope. And we have also recorded the different developmental stages and published in the journal. What stage it reaches 2 cell, what stage it reaches 4 cell, 8 cell, blastula, gastrula, morula, and when it is hatching, and when it is absorbing the yolk sac, we have also recorded. And this is the yolk sac larvae, and this is around maybe 15 days. This is 1 month old fingerlings. So we have standardized this all technique. We have standardized in St. Xavier's College, Palankote, very old, 20 years before. So this is the hormone method, intramuscular injection. Oprim we have used, and we have also standardized 4 different hormones: carp pituitary extract, human chorionic gonadotropin, oprim, and overtime. So we found that 50 mg of carp pituitary extract is optimum for induced spawning. Similarly, for human chorionic gonadotropin, 2000 international unit per kg body weight. Oprim and overtime, 0.4 to 0.5 ml is recommended. It's a good dose. We did not see any differences in 0.4 and 0.5 ml. Right? So one vial it comes with the 10 ml, and it is around 500 rupees. So why we need to go for higher dose? Even 0.4 ml also do perform very well. So the recommended hormone dose is 0.4 ml. And this is the way we need to identify the male and female fishes. The female fish is somewhat very bulged abdomen. And this is a female fish, somewhat very bulged abdomen. Genital papilla reddish in color. Whereas in the male fish, the genital papilla is very, very minute and little pointed. And if you have the hand lens, we can easily identify the male and female fishes. And then to identify the healthy female fish, you can also give a slight pressure in the abdomen. Eggs will freely ooze out. We can also observe under the microscope the readiness of the egg, the readiness of the female for induced spawning. Additionally, we can also test the maturity of the female. We can also confirm by slightly pressing the abdominal cavity. Eggs will comes out very freely, then we can observe under the microscope. So this is a breeding set. One female and 2 males. And this is a female fish, very bulged abdomen. So the female is good for breeding. So hormone administered 0.4 ml to 0.5 ml per kg body weight. And then After injection, male and female fishes need to be released into the earthen pond. And within 24 to 30 hours, we could see the the releasing of eggs. Fertilization takes place. So we have conducted the study in St. Xavier's College. We tested with the different hormone doses, 0.2, 0.4, 0.6. And we found that 0.4 and 0.6 was found to be similar responses in terms of fertilization, 98% fertilization. And after fertilization, it takes 23 to 24 hours for incubation period, hatching. How much percentage? It is a 82 to 91% we have got hatching. And again, 0.4 and 0.6, we did not see any significant differences in hatching rate. So that is what we recommend. 0.4 ml is viable optimum dose for induced spawning the snakehead fishes. And we also standardized very old, this one, 2001. And we have used this kind of HAPA method. And this is my first research article in St. Xavier's College. We have standardized it. And along with other fish species, the same earthen pond, you can also construct like this, install like this, cages. After hormone administration, you can put them inside. It is easy. Small scale farmers, this technology, the low cost breeding method is recommended. And we have also published the embryonic and larval development in the journal Taiwania. And we have recorded from fertilization until hatching, the color changes, morphological changes, when it reaches 2 cell, 4 cell, 8 cell, gastrula, morula, at different stages. When it opens, the internal organogeny starts. When it starts feeding, right? So this is a fertilized egg. And fertilized egg, only one egg, observed under the microscope. And we have a morula stage. And this is 6 hours old embryo. And this is 24 hours old embryo. We could see anterior and posterior. Can you see here? This is a head region. And this is a tail region, right? And it is about to hatch 24 to 25 hours. So the tail wagging will be there and beat the the outer layer. The cell will burst and young ones will come out. So this is the hatching. Can you see the outer layer and the tail? And this is a yolk sac. The first 48 hours, we no need to feed them. They can utilize the yolk as a nutrient for them, right? And this is a ANS, 4 hours old, observed under the microscope. And this is a hatchling of Channa striatus in different stages, 16 hours, 8 hours, and 36 hours old larva. And this is 10 days old. So at the early stage, we have standardized some zooplankton, Artemia nocli, moina, and then semi-moist diet like chicken waste, fish waste, we have standardized. And that can be recommended for. They are, frankly speaking, they are carnivorous. So they prefer some kind of animal protein-based feed. So nowadays, after 1 month, they do accept commercial feed. Commercial feeds also available in India and Malaysia. So during my time, during my PhD time, we have standardized a semi-moist diet using chicken waste and fish waste. Like noodles we prepared and we have given them as a feed. So we found that was healthy and we produced a good number of fingerlings, and also we have supplied to the farmers those days. So this is early days, they are very colorful, ornamental value, early days. So this is 1 month old. This is harvested from only one female fish. And then another fish species also we have standardized the breeding method, snakehead fish, spotted snakehead. In Tamil we used to call korvamin. This is also very highly edible fish. It reaches 300 gram maximum. The same methodology we have adopted. Identification of male and female fish, 2:1 ratio, intramuscular injection. And it also similar to the other hormone, other fish species like snakehead fish, Channa striatus. And same dose, 0.4 to 0.6 is recommended. And slightly the incubation period is different. The hatching rate also different when compared to the other species. And using overprim, the another study we have published in the journal Asian Fishery Science. I have already mentioned, even after hormone administration also, the male and female fishes, they do protect the young ones. Can you see here? This is egg mass, and this is what you call the male fish. They are very, very aggressive. Male and female fish always with the fertilized egg. They will safeguard the eggs. They will protect the eggs. And see here, the small dark dots is what you call the fingerlings. And this is a parental fish. Always they safeguard. So we found that male fish are very comparatively very aggressive than the female fish in the particular fish. So first time we have reported in this particular work in the current science. We have observed the complete the spawning and the breeding behavior and reported in the journal current science. So next one is about catfish. So the breeding method somewhat different than the snakehead fish, Channa striatus and the spotted snakehead. African catfish, we followed differently, artificial fertilization. And also this is very delicious fish species in Malaysia. High market value next to tilapia. And the high yield, they are carnivorous. They are also cannibalistic, drawback. They're very hardy fish. And they can be cultured in what you call low dissolved oxygen water, even sewage water. This also can be cultured in paddy field, that much hardy fish. But especially in Malaysia, the Malaysians, they like the catfish like anything. And that too, they prefer 200 gram to 300 gram size. Whereas in Thailand, the catfish used as a fish ball. They grow even 2 kg, 3 kg catfishes, 2 years old. They use the meat for fish ball and fish fillet, right? And this is my undergraduate student. So the breeding method somewhat very, very easy compared to snakehead fish. Very easily you can identify the male and female fish, abdomen, and this is a female fish. The genital papilla somewhat very broad, whereas the male fish, the genital papilla pointed, very, very easy. So during the reproductive season, the genital papilla somewhat very reddish in color. The male is good for artificial induced spawning. And this is a female fish, the abdomen somewhat very broad, and this is a male, the abdomen somewhat very tapery, slender. Intramuscular administration, we used 0.4 ml per kg body weight, overprim. And after hormone administration, the male and female fish we need to keep separately. Male and female fish need to be kept in separate system, separate tanks. We allow 24 hours of latency period. Right? And after 24 hours, the female is ready for collecting the eggs. And can you see the abdomen, somewhat very bulged, swollen abdomen? And after 12 hours, you can easily collect the eggs from the female. The eggs will come like a very free flow. Collect in a what you call clean container without any much water from the female fish. And this is the eggs were collected from one female fish. Maybe around 100 grams, 1 kg fish, that much quantity of eggs will be there. And then male fish, after collecting the eggs from the female fish, male fish somehow we have to sacrifice. Reason, the collecting the sperm from the catfish very, very difficult. So we need to do small dissection, take out the testis from the male fish. We need to take out the testis from the male fish, and then we need to cut into small pieces. And this is the testis were collected from the male fish. We need to cut into small pieces using the scissors, then collect the milk. So inside the testis, please take note, the sperm is immotile. They will not be active. When they reach the aquatic or water, the sperm will get the motility. That motility also will last for few hours. Sorry, not few hours, few minutes. With that period, the sperm need to fertilize the egg. If not, the sperm will die. So the whitish fluid is the milk. So once you have collected the milk, we need to activate with water so that the sperm will get activated. Then you mix the collected sperm and the egg. And then we need to add some more water to activate the sperm so that the sperm will be very active and penetrate the egg and fertilize the egg. So within 5 minutes, the eggs will be fertilized. Then what we need to do, we wash any excess fluid which are present with water, excess water. 1 or 2 times we need to wash, then we need to go for incubation. So incubation, we can use it any aquarium or swimming pool kind of small things, or any containers or any cement tanks. In our laboratory, we used one swimming pool kind of thing. I will show you later. You feel a little fun. So this is the fertilized eggs incubated in glass aquarium. And we have also recorded the different developmental stages under microscope and published in a journal. So 2-cell stage, this is a fertilized egg. 2-cell, 4-cell, 8-cell blastomeus stage, 16, 32 cells, and 15 somites, 20 somite stages. And this is just before hatching. Can you see here this outer layer? And this is at hatching, what happens. And then we have also recorded until the yolk sac absorption. So this is a yolk sac. And this is what we call anterior region. Eye vesicle is not yet developed. The fins are not yet developed. Barbels not yet developed at this 5 hours old hatchling. So after 48 hours, the yolk completely will be absorbed. Can you see here? This is the anus. Mouth also not yet fully developed. The furrow is there, but it's not yet developed. So once completely the yolk sac is utilized, then we need to have a what you call their feed. The live feed should be ready. Normally we use Artemia napoli. We collect the what you call, we purchase the Artemia cyst and we hatch it in our laboratory and we use as a feed for catfishes. And this is 20 hours old. We could see the barbel, this is a catfish. And we could also see the what you call the color changes. And this is a yolk sac. Still yolk sac is there. 20 hours. Can you see here the yolk sac size is reduced? And this is a what you call GIT, digestive system. And this is the anus. And mouth also developed. We could see the color changes. We could see the vertebrae also. Right? And then 3 days onwards, you can start giving a feed. They can also eat Artemia napoli as well as Artemia cyst. And very, very easy. The high survival rate also you can observe in catfishes. So in my laboratory, we used a small swimming pool kind of thing. Children's what you call the swimming tub. We can use it like this. So this is my undergraduate student. We use this kind of small pool, maybe even 1 meter diameter. And water level we used to keep 30 centimeter, not more than that. Right? So Artemia napoli we used to give, and after what you call 15 to 30 days, we start giving the what you call pelleted feed. So catfishes, they do accept the pelleted feed. Even what you call sea bass starter feed, prawn starter feed, we used to buy and use it for fingerling raising. Right? So they're very, very aggressive. You can also go for high density fingerling culture in this kind of tanks. Very, very simple, the technology. And then we also used to give some kind of training for local farmers. And this is my student. And a couple of farmers we have trained in our laboratory. And sometimes we used to go to the farms itself and we conduct, and we take our students for farming facility to learn the breeding technique. Right? We take them there. And the whole methodology, the reproductive, the breeding and seed production technique for the catfish we have developed in our laboratory. We also published in one what you call journal. This article has been published in I think 6 different languages. 6 different languages this article has been published, and also very good citation as well. And the breeding method technique we have also published in this journal. We found that similar to other fishes, hatching rate even 0.4 ml were higher. Higher doses slightly lower, 93% hatching rate. And fertilization, there was no such differences between 0.5 and 0.6. Slightly what you call low dose, the fertilization also higher. But 0.4 and 0.6 statistically, no differences. That's it, participant. So I would like to thank my professor, my supervisor, my guide, Prof. Hanifa. Without him, I'm not there. So he funded me and he supervised me. He gave me this life. And also I'd like to thank my postgraduate students, my undergraduate students, AIMS University, Malaysia, for supporting me and doing research under my supervision. So thank you so much, participant. If there is any questions, I'll be happy to answer. I have taken more time. Sorry for it. If there is any questions, I'm there to answer. Most welcome. Dr. Alagraj, over to you. Participants, do you have any questions? Participant, do you have any questions? Any questions? Dr. Alagraj, are you there? So I have done it. If there is any questions, I will be answer. If not, I will leave this the session. I hope next also we have one more talk. Any questions from the audience? No questions?
Hello, sir. Sir, good evening.
Any questions? If not, thank you so much for giving this good opportunity. Thank you.